Greetings, everyone across the globe. Welcome to the final day of the SynCell2020 virtual symposium. This is the International Conference on Engineering Synthetic Cells and Organelles. Next slide, please. My name is Gabriel Lopez. I am chairing this conference along with my co-chair, Joachim Spatz. I am Vice President for Research and Professor of Chemical and Biological Engineering at the University of New Mexico. And we have a great program today. Next slide, please. Very quickly, here is the rundown for today's schedule. We are hoping to start with Drew Endy as a keynote. He's from Stanford University and we'll be talking about upwelling, challenges arising from the bottom of the Earth's life well. I think we have not heard from Drew yet, and so we may have to make some adjustments if he's not here in a few minutes. Next, we will have a tutorial on synthetic building blocks to grow functional tissues from Dr. Laura Delaporte. And she is from Leibniz Institute for Interactive Materials in Aachen. Then another tutorial, liquid-liquid phase separation of intrinsically disordered proteins by Nick Carroll. And then our program highlight today will be on the Build-A-Cell Consortium. This will be given by Kate Adamala at the University of Minnesota. Then we will have a panel discussion on the future of synthetic cell technologies for mitigation of viral pandemics. And the panel will be Andy Ellington from the University of Texas, Austin, Kate Adamala from University of Minnesota, Eberhard Bodenschatz from Max Planck Institute for Dynamics and Self-Organization, and Michael Grunze from the Max Planck School Matter to Life. And then there will be some final remarks that I will talk about. The moderator today will be Andy Ellington from University of Texas. Next slide, please. Just to remind everyone how to participate, you can submit questions using the Q&A button at the bottom of your screen. And you should keep an eye on those questions. Everyone should keep an eye on those questions and upvote the questions that they think are particularly relevant and poignant. The number of upvotes will help determine which questions the moderator will choose to ask. And then finally, you can see who's here at the top of your screen, how many people are joining. Next slide, please. Now for some acknowledgements. I just want to thank people and organizations that have helped bring this virtual conference to fruition. The University of New Mexico and Max Planck School Matter to Life were the primary partners in this. At UNM, I'd like to acknowledge the hard work of Ms. Emily Ballo, Dr. Mary Jo Daniel, and Pamela Garcia Ramirez, who have all worked for several months now to put this together. We also have a distinguished organizing committee of about 10 people who have helped put together the program, the dissemination, and fundraise. And then finally, I'd like to thank all the speakers, panelists, and moderators, as well as these organizations who have provided support for this conference and these activities. Next slide, please. Actually, one more very important acknowledgement, and that's to our technology enablers. This virtual conference was really the brainchild of Andrew Hole and Jennifer Young. They are postdocs at the Max Planck Institute for Medical Research, and they are behind the scenes running everything today. Next slide, please. I'd just like to remind everyone that this virtual conference was initially scheduled as an in-person conference for this May 2020, but we had to cancel that and postpone it till next year. So we still intend to hold the in-person conference in spring 2021. So please be on the lookout. Keep your eye on http://syncell2020.unm.edu. We will be sending out routine reminders of this as well, and we hope to see you in Santa Fe. Next slide. With regards to the upcoming conference, in-person conference, please feel free to send feedback and suggestions regarding speakers to SynCell2021@unm.edu. I was told yesterday that this email might actually not be active yet. We'll try and get it active today. So either SynCell2021 @unm.edu or SynCell2020, both of them should work just fine. But if one doesn't, please just use the other. Okay, next slide, please. All right, so at the end of the day today, at around 11:35, we'll have some final remarks, and usually at the end of an in-person conference, we take a group photo to commemorate it, but obviously we need a virtual workaround this time. So at the end of today's meeting, we would like to take a virtual group conference photo similar to what you see as an example on the screen, but hopefully much larger if we can get everyone to participate. We cannot do this in the current webinar format of Zoom since attendees cannot turn on their videos. Therefore, we have to switch to a regular Zoom meeting format in order to do this. We've sent the new Zoom link at the bottom of today's reminder email. So please join that link after the meeting ends. Since we can only host 100 people on a Zoom meeting at a time, we have designated times when photos will be taken, listed here on the screen and also in your reminder email. So if the meeting is full when you try to join for the photo, just try to join at a different time as we clear people out. Through this methodology, we hope to compile a picture with large numbers of participants. We're reflecting the large number of participants. We'll compile that whole picture and then put it online on our website, but also save it for posterity and use it in demonstrating the power really of this virtual format on a global scale. So please participate here, and we will really appreciate if you can help us with this task. I will give this information again at the end of the day. So without further ado, then I guess I will turn this over to today's moderator, Dr. Andy Ellington from the University of Texas. Thank you, Andy.
Thanks, Gabriel. I assume everybody can see and hear me at this point?
Yes.
You're still front and center in my screen, Gabriel. Here we go. Am I coming up? I'll just go ahead and start pattering.
You are.
So we have a great session today. I'm going to patter for just a second in case Drew manages to get his internet problems fixed and can show up on time, and we'll keep the rotation the same. But one of the things that I find most fascinating about this conference, and again, a great job by both the New Mexico crew and the Max Planck crew for putting this together, but it really overlaps with three of the fields that I like to associate myself with, at least. One of them is synthetic biology, and that's probably apparent to many of you. It will be very apparent with Kate Automala's talk later today. But the whole notion of being able to do bottom-up rational construction of any of a variety of biological devices, obviously the synthetic cell would be sort of a next milestone. We've already been able to construct genes, gene circuits, genomes, but so far, in terms of actually making a cell from scratch, that remains, sort of something that we can aspire to. In addition, this field overlaps nicely with what used to be called Origins of Life, but in the more modern era is called astrobiology. How the original cells came to be on the planet Earth is, and elsewhere, is something that is open for debate and has been for literally hundreds of years. And nicely, again, the field of making synthetic cells provides an experimental counterpoint. Just like when you were able to synthesize or do directed evolution of ribozymes, you understood something about the RNA world, even if you couldn't have been there for its early onset. When we can go to the point of synthesizing cells, I think we'll understand much more about life's history. And finally, I think there's a strong overlap with the nucleic acid nanotechnology community. We saw this with Darko Stevanovich's talk yesterday. But in addition, there's an element to nucleic acid programmability that is very special. And that's one of the reasons I like interacting with that community. Is it is by far the most rational design community of biology, in a sense, in that you can go from simple base pairing up through entire computations. And there's also a mystery, in a way, in terms of how the first cells came to be, between sort of the analog computation that's inherent in a cell wall and cellular division, and the more digital computation that's inherent in the genome that's present inside a cell. And so it's both a counterpoint and informs how we'll do potential rational design of synthetic cells into the future. Okay, with that, I'll sort of quit pattering for a moment. I'm not sure Drew has managed to fix his problems yet, but that's just sort of how I see this field, the synthetic cell field, fitting in between other fields that have maybe grown up a little bit earlier, but now support and overlap with what's going on here. With that, I'm going to give a brief introduction to Nick Carroll. Nick is an assistant professor in the Department of Chemical and Biological Engineering at the University of New Mexico. His primary research focus is programming soft materials as the building block level for self-assembly into functional colloidal architectures. Nick's postdoc work at Harvard focused on material science with a concentration on microfluidic-based smart microcapsule synthesis. This multidisciplinary work was in close collaboration with Kari Jensen, a Harvard postdoc, and Professor Holbrook's group through the MRSEC droplet templated materials area. And with that, I turn it over to Nick. I'm very much looking forward to your talk.
Okay, great. Thank you very much. Let me see if I can do the share screen with folks. And I think that's working. So can everyone see my screen and hear me?
Yes.
Okay, very good. Thank you very much, Andy, for the introduction. Today, I will be giving a brief tutorial on the concept and the kind of technologies that underpin liquid-liquid phase separation of intrinsically disordered proteins. So the outline for the tutorial, which essentially you can think of it as kind of a starter pack for folks who might be interested in doing research or moving into the field of phase separated intrinsically disordered proteins, particularly through the lens of self-assembly of ribonucleoprotein structures to essentially replicate or create synthetic organelles, which I'll talk about. So the outline for today is first an introduction to intrinsically disordered proteins, kind of a quick introduction to their role in biology and to some of their physical and chemical nature. Then I will go over phase diagrams, in particular IDP solution phase separation, so kind of an introduction into the very basic thermodynamics that underpins liquid solution phase separation. I'll then talk about how to handle multi-component protein mixtures in terms of design and synthesis. I will talk about how the field is moving very quickly into creating synthetic phase separating ribonucleoproteins, so creating complexes of RNA or DNA with intrinsically disordered proteins. And then I will give a brief conclusion slash outlook on what I think the future of the field will be, or at least the direction that it's moving in. So intrinsically disordered proteins flew under the radar for quite some time, primarily because I think they have a rather boring appearance in that they're just random coils, so they lack secondary structure. They lack these origami shapes that we typically associate with proteins. So what defines an intrinsically disordered protein, in addition to this random coil configuration, is they're characterized by a typical sequence, which is proline and glycine rich. And so proline, which is structure breaking, kind of works to prevent these proteins from folding and achieving secondary structures. And then the glycines are essentially very flexible linkages in the peptide. And so this allows them to have this kind of random coil nature. So as you can see on the left, this is just a representative diagram from the human fuse protein, which comprises alternating or concatenated intrinsically disordered regions, along with ordered regions that are responsible for binding to specific types of RNA. So many of these domains that bind to RNA are themselves also disordered, such as RGG1, RGG2, and RGG3. And what's interesting about these domains is, in addition to maybe large regions in these native proteins that are intrinsically disordered and can induce phase separation, disordered regions which bind nucleic acid can themselves undergo phase separation. And so it's been discovered that if you either recover these proteins or you synthesize them, in vitro, they have what's known as an upper critical solution temperature behavior, or UCST. And that is at large temperatures, these proteins are soluble, but as you cool below some transition temperature, they undergo a phase separation as drawn out in the schematic on the lower right-hand corner here. And so when the protein is in the soluble phase, this represents a good solvent. This is in the random coil self-avoiding walk configuration. And then below a so-called theta temperature, and theta is where the polymer is essentially acts as an ideal solution. This is when phase separation occurs. And so these proteins will collapse into what's known as a globule. These globules then become, they act like de facto oil drops, oil and water. And so they will coarsen or coalesce with one another and form a protein-rich phase. And so what's interesting about this, about these proteins is, as you can see, this so-called Ponder score, which essentially represents the level of disorder. And we see that some of these RNA binding domains are also disordered. Well, this maps on very well to this so-called plaque score, which is related to prion behavior. And the higher the score, the more likely that the protein will undergo a liquid-liquid phase separation. And so we see that this kind of disorder maps on very well to this propensity for phase separation. So why did biologists and scientists in general become interested in these intrinsically disordered proteins? Well, this is because the cell uses these intrinsically disordered proteins to assemble a host of phase-separated organelles, which are called membraneless organelles, in both the cytoplasm and in the nucleus. And so the trigger for this type of phase separation, which clearly physiological conditions are going to be isothermal, are things like subtle changes to the chemical composition through methylation and phosphorylation, for example. Those are two very common kind of triggers for phase separation. Also, these phase separating proteins can concentrate around other scaffold molecules, nucleic acids and other proteins. And so the cell will use concentration to trigger phase separation. And finally, we would expect that there would be many multivalent interactions between intrinsically disordered proteins as well as nucleic acids. And so many times these types of interactions can trigger phase separation. So two of my favorite so-called membraneless organelles, one is in the cytoplasm. These are known as stress granules, as you can see in the bottom corner here. And what's interesting about stress granules is when our cells undergo some type of environmental stress, such as heat stress, for example, even viral infection, these proteins will activate and will bind particular RNA. So in the case of heat stress, for example, they will sequester RNA that is not necessary for long-term survival. When the stress subsides, these granules will then resolubilize and release RNA that can then be expressed and could serve important functions for long-term survival. What's also unique about stress granules is in the case of harmful nucleic acids and so forth, there's a decision-making process that occurs within the stress granules. These nucleic acids can be passed to other types of granules, and they can be cleared out of the cell, for example, through autophagy. So my other favorite type of membraneless organelle is known as the nucleolus. And so truth in advertising, this exists in the nucleus of the cell. And what's very interesting about this membraneless organelle from a material science perspective is that the granule will form a multi-layer configuration. So each of these protein layers are not only immiscible with the surrounding nucleoplasm, but they are also immiscible with each other. And so coming from a polymer background, I was very interested in how the cell could potentially assemble these multi-component structures. Okay, so what else is interesting in terms of the function is it's thought that each layer is responsible for a particular step in the ribosomal assembly process. So the type of intrinsically disordered protein that we've been working with over the last few years, in particular myself and Gabriel Lopez, are known as elastin-like polypeptides, or ELPs. So they have a very low complexity, minimal repeat sequence. It's a pentameric repeat of valine, proline, glycine, X, which can be essentially any guest residue, and glycine. And so these exist as random coils. And as we discussed in an earlier slide, they satisfy this criterion of being rich in proline as well as glycine. So these particular polymers are lower critical solution temperature polypeptides, in that below some transition temperature, they're fully soluble, exist as random coil. However, upon heating above the transition temperature, they undergo a phase separation, form these so-called thermoglobules, which then coarsen and form a protein-rich phase that is surrounded by a supernatant phase. And so this diagram represents turbidity measurements that shows the transition temperature as a function of concentration or volume fraction. And so the larger the concentration or the volume fraction, the lower the transition temperature, at least in the dilute branch of the phase diagram. So as I mentioned, native proteins are typically show UCST phase behavior. So there's a really nice work that was that came out a few years ago from the Chilcote group that essentially assigned heuristics for how to use charged and polar residues to create minimal repeat intrinsically disordered proteins that can exhibit either lower LCST phase behavior or UCST phase behavior. And so this kind of provides a rich menu of heuristics so that in designing synthetic membraneless organelles, for example, one does not have to necessarily use ELPs or LCST polymers. The amino acid composition can be tuned in such a way so that they can exhibit UCST phase behavior, which is more representative of native proteins. So why do we use ELPs as a model IDP? Well, one of the reasons for this is that the cloning process as well as the purification process is relatively straightforward. For cloning, the kind of canonical process is known as recursive directional ligation by plasmid reconstruction, which was also came out from the Chilcote group. And this is essentially just a very clever way to cut and concatenate expression vectors so that oligomeric inserts can be inserted in a stepwise fashion. So you basically cut these plasmids, you then join them together in very precise locations with so-called sticky ends. And so as you repeat this process, you're able to make these codons that encode for these multiple repeat polymers. And so this is exemplified in the schematic. Because of the nature of the phase separation, the LCST behavior of ELPs, they can easily be purified using thermal cycling. So after expressing these proteins in bacteria, you essentially lyse the proteins, and then you can use temperature to either form to cause ELPs to form in the aggregate phase or the supernatant phase. And so you can alternate between these in this so-called thermal cycling and use centrifugation steps to separate the ELP from the cell lysate. And so these ELPs can be purified at relatively large quantities, hundreds of milligrams up to grams. And so it makes it quite amenable to use as a biomaterial. So the most basic kind of physics that underpins this type of liquid phase separation is a mean field theory given by Florey Huggins. And so in this free energy expression, you have the first two terms, which are purely statistical, and these represent the entropic components of the free energy. And then you have a last term, which is essentially the thermodynamic energy. So the phi represents the polymer volume fraction. N, which appears in the entropy term for the number of ways to configure the polymer, represents the degree of polymerization. And then in the energy term, we have this chi interaction parameter. And so this chi interaction parameter essentially describes the energy penalty for exchanging homotypic interactions for heterotypic ones. In other words, exchanging, for example, polymer-polymer interactions for polymer-solvent interactions. And so this first term, which is the entropy, the configurational entropy, always promotes mixing, and we see that that scales as 1 over the degree of polymerization, or the chain length. And so the way that I often like to describe this to students in my thermo class, and I'm dating myself a little bit, is maybe we recall or know of this game called Snake, which often appeared on calculators and so forth. And so as the game proceeds, this snake, or this thing that looks like a polymer, continues to grow in chain length. And the game becomes increasingly difficult because there are less ways to arrange this growing polymer in this volume. And so this represents the energy penalty for having a large polymer. So in other words, the configurational entropy is decreased as the degree of polymerization increases. And so all of these phis, in particular the phis that are multiplied by this interaction parameter, well, 1 minus phi, if we're talking about just a single component polymer solution, would be the volume fraction of the solvent. And so this just represents the probability of a solvent and a monomer in the lattice interacting with one another. And so below some critical chi, the polymer is soluble and exists in the random coil configuration. And so in the free energy diagram, as a function of volume fraction, this is represented by the black curves, which we see are all convex. So there are no instabilities in the free energy curve. However, upon surpassing a critical chi, the polymer phase separates, and we see now that we observe these concave regions, which begin to form in the free energy curves. And so this is when phase separation will occur, and we form these two phases, a polymer-rich phase and then a solvent-rich phase. So we can then map the phase diagram by just taking the partial derivative of the free energy, of the Florey-Huggins free energy, and finding a common minima, which is given by the first derivative of the free energy with respect to volume fraction in the dilute region, which is given by phi prime, and in the concentrated region, which is given by phi double prime. And so, of course, we understand that a change in free energy with respect to material represents the chemical potential. So when the chemical potential of these two phases is equal, we can then essentially map that each of these points as a point on the binodal. And so each of these curves represents a unique chi. And so if we're using temperature as the trigger, each of these would represent the chi at different temperatures. And so I just mapped on in red to kind of make clear the binodal curve, which separates the one phase from the two phase region. Then there's another important construct within the free energy curves. This is known as the spinodal curve. And so this is just given by these inflection points within the free energy curve. And the spinodal represents phase separation where there's no energy barrier, essentially no energy barrier. So phase separation occurs homogeneously throughout the solution, whereas outside of the spinodal, but still within the two-phase envelope, this represents a nucleation and growth regime. So what does this phase separation look like? So what we typically do, and I think one thing that's particularly useful in terms of looking at these types of structures, providing a window into them, is to use microfluidics to create water droplets, which contain the IDP or the ELP solution. And so what this thing looks like is below, whoops, let's see, I'm just gonna play. So below the transition temperature, the droplets are clear because the ELP is soluble. And then upon heating into the two-phase region, we see that that the ELPs come out of solution, form these thermoglobules, they coarsen, and when phase separation completes, we observe a polymer-rich coacervate in the center of the droplet surrounded by a water-rich shell. And so this is the other kind of advantage that's provided by knowing the binodal, and that is at some temperature where phase separation occurs, the intersection with the binodal represents the volume fraction of the protein in the coacervate phase, as well as, and then the other intersection gives us the volume fraction of the protein in the supernatant phase. And so there are multiple ways to measure the binodal. One involves dynamic light scattering, which is kind of a painstaking process. What you do here is you just make up a bunch of samples at different volume fractions, and then you heat very slowly, and you observe a change in the dynamic light scattering properties. And so you kind of mark each of these down. A way to measure the spinodal is in the soluble state, you make up, again, ELP solutions at different concentrations. You then take static light scattering measurements, and you can plot the temperature versus 1 over the intensity. And so you extrapolate this back to essentially an infinite scatter. So you extrapolate to the y coordinate. And this essentially represents a point where you have an infinite scatter, or your entire volume scatters. And so if you do these for various concentrations, this allows you to map out the spinodal diagram. The spinodal curve. So this is for a single component. What about multi-component ELPs? So what we've been able to do is kind of just extend this very simple Flory-Huggins theory to describe multi-component mixtures, where we're interested in the structure of the assembled ELPs in the coacervate phase. And so, for example, they can form core shell structures where the two ELP phases are immiscible with one another, or they can form mixed coacervates where the two ELPs will mix. So just a quick demonstration of what this looks like to show that so we take two ELPs, they just differ subtly in their guest residue composition. And what we do is we heat these droplets under the microscope above the transition temperature of both droplets. We see that the blue labeled and the green labeled ELP come out of solution at the same time. However, we don't observe them mixing with one another. So then we cool below the transition temperature of the more hydrophilic green labeled ELP. We then heat above the transition temperature once again, and we observe that these ELPs will form these core shell structures, where they're immiscible with one another. And so the way that we can handle mapping out these types of phase diagrams is simply to add in a few more expressions into the Flory-Huggins free energy. So we want to add in an expression that accounts for the entropic contribution of a second polymer, polymer B, to the free energy. We then add another energy term, essentially, that describes the interaction of polymer B with the solvent, in this case, water. And we now have a new interaction parameter, which is not required for a single component ELP mixture. And this is known as the cross polymer chi. So this is essentially the energy penalty for polymer A interacting with polymer B. And so if we plot these free energy surfaces, for example, if we plot just the entropic terms, we see a convex shape that forms throughout the free energy surface. If we plot just the energy terms, we observe this concave formation, which represents that, these energy terms always kind of work to promote demixing. And then if we plot a combination of these, we have these free energy surfaces with various curvature where we can predict whether or not we will have a mixed coacervate, which represents a two-phase system. So in other words, two proteins that are in the coacervate surrounded by a water-rich phase, or we have three phases where we have polymer phase 1, polymer phase 2 surrounded by a water-rich phase. And so the way that we can map out the phase diagram then is analogous to the single component. We can just look at inflection points at the curvature. And so what we do then is we just plot the curvature of the free energy expression as a function of volume fractions of the two polymers. We look for the points that pass through the zero curvature plane, and these essentially project onto a phase diagram that allows us to predict whether or not we will form mixed or core shell coacervates. So in the case of mixed coacervates, we can take two proteins which are chemically dissimilar but have a very similar phase behavior. And what this represents then is a very low cross polymer chi. And so in these cases, even if we have two polymers which have rather large interaction parameters with the solvent, because they have a low cross-polymer interaction, we never observe the appearance of a three-phase region or a core shell region in the phase diagram. Conversely, if we have two polymers which are chemically dissimilar, but they have a large interaction parameter, we observe the formation of a three-phase region in the ternary phase diagram. And so this is just a qualitative example that I put up here where we just have two chi values that we've assigned to polymer A and polymer B with water. And then as we sequentially increase the value of the cross-polymer chi, we see the appearance of this three-phase region, which is given by this other kind of triangle shape that forms. Then what you could do is you take these phase diagrams can then be projected onto a volume fraction equal to 1 plane. And this gives us the ternary phase diagram that we typically think of as canonical chemical engineering phase diagrams. So an interesting thing to come out of the work is the effect of chain length. So you can take two ELPs which have a relatively large cross-polymer interaction parameter, and for a protein which has a rather large chain length, 160 repeats, we observe a core shell formation. The core shell formation is given by an operating point in the three-phase region, but by truncating the polymer from 160 repeats to 40 repeats, we observe mixing of the ELPs in the coacervates. And so this reduction in the entropic contribution from the polymer corresponds to a shrinking region, a shrinking three-phase region where core shell structure will occur. And so by maintaining the same operating point, we can use chain length to modulate demixing or mixing of these proteins in the coacervate. And so this is something that we're working on at UNM to kind of modulate chain length in situ by controlled concatenation or truncation of these polymers. And the idea is having this predictive phase diagram, we can move around the phase diagram and potentially have layer mixing and demixing. And so this could be very interesting for potentially controlling chemical reactions in synthetic environments. And so there's a MATLAB code that we've written to kind of, allow us to plot and to observe these phase diagrams. So it's a relatively, it's not really a sophisticated program, but for any students who might be interested in this, I would be happy to send along this code to you. And it's quite useful to mess around with these chi parameters and chain lengths and so forth to see, how they affect the polymer behavior in the coacervate. So finally, we extended this by adding another immiscible ELP to form a 3-layer coacervate. And so at least from a structural perspective, this allowed us to move a bit closer to the multi-layer configuration and phase behavior of the nucleolus. So the next phase of the work is to create phase-separated IDP nucleic acid associated complexes. And so in particular, folks are interested in recapitulating the behavior of cellular IDPs. In that, cellular IDPs can bind to specific RNAs with particular sequence as well as structure. Also, it'll be important to understand and to map out the phase behavior of proteins in the presence of nucleic acids. And so these are just some representative images showing RNA binding domains from the fused protein. So this is zinc finger, which binds to RNA. These squares just represent nucleotides. And then this shows a fused RRM, which is an order domain that is bound to a stem loop RNA. And so in work that's been done, it's been found that various domains, various RNA binding domains have propensity to bind to particular sequences or structure. Poly-A tails are another type of structure. So this is just showing the protein structure, the 3D structure of these proteins. RGG3, which is largely disordered, itself can undergo phase separation, and then RRM has kind of a larger number of these ordered domains. And so, investigating protein phase separation in the presence of RNA is important because RNA also drives separation by acting as a scaffold for multivalent RNA binding proteins or by participating in intermolecular RNA interactions. And so interestingly, RNA itself can phase separate and form its own phase. So this is work that Gabrielle and I have been doing at UNM. We have an ELP protein which can undergo a configurational change and bind to DNA. We demonstrate this in droplets. So in this particular example, the first column is just Brightfield, which shows the phase separating ELP. So it spinodally decomposes, forms the protein-rich granule. The middle column shows the DNA, which is labeled red. So DNA is the only labeled phase. And we see that we have efficient capture of the nucleic acid within the protein coacervate. And then this is just an overlay. And so this is the work of our PhD student, Telmo Perez, who has really pushed these ribonucleoprotein experiments and kind of understanding forward, and particularly in the last year. Also, we've taken these ribonucleoprotein granules and we've demonstrated that we can form them in sequestered nucleic acids in droplets which comprise not just buffer, but these droplets are comprised of cell-free TXTL extracts. And so this shows the promise for using these in synthetic constructs. This is particularly important for us because we were lucky enough to have this work funded by the NSF CBED through the Rules of Life, through a Rules of Life eager grant. And so I'm working with Matt Lakin, who's also at UNM, so that we can demonstrate controlled metabolism or controlled expression of RNA in synthetic cells by either sequestering or releasing the RNA akin to how stress granules operate in the cell. So other important work that's been done in terms of creating these proteins which can also bind nucleic acids comes out of the Chakotay lab. And so they have created a fusion protein by concatenating an ELP to an RGG domain, ostensibly also from the human fuse protein. And what they've been able to do is to sequester RNA in these ELP coacervates so that they make the RNA either available or not available for expression. And so we see that both regions, ELP and RGG, are relatively largely disordered. This bottom figure shows the ELP fusion binding to RNA and sequestering it within the coacervate. So this is also in water droplets. And then this slide demonstrates that they were able to sequester the RNA within these granules. So the background that you see here, so these are for, this is for minus IDP. So this is just the TXTL expressing a green fluorescent protein. This is the just the ELP portion of the fusion protein that lacks an RNA binding domain. And in both cases, we see green increasing because the TXTL system is expressing the fluorescent protein. However, notice that for the fusion protein, which has the RNA binding domain, this RBD, there's not a large degree of green fluorescence that forms in the droplet. And so this represents the blue curve in this plot, which is essentially the signal from the green fluorescent protein, and then this is time. And so using this, they've been able to effectively turn off or turn on the expression of this green fluorescent protein RNA. So Outlook, other things that folks have been working on and focusing on are ways to characterize the thermodynamics of the system in a very quick fashion, potentially to use things like image analysis to gain insight into the thermodynamics. And so this is work from Adam Quintana, who's a graduate student here at UNM working in the Lopez lab. And in these examples, he's modeling spinodal decomposition of multi-component systems. And what's very cool is we can track the phase separation through this phase diagram that he has coupled to this spinodal decomposition simulation. And what we observe is that we have two phase separating Florey-Huggins molecules essentially, and then we have another phase, which is the solvent-rich phase. And so this is recapitulating the things that we see experimentally in these 2D simulations. Other work that he's done recently, very recently, is he's been able to take two different polymers. They have a rather large interaction energy between them. And what he's been able to show is that akin to the wetting behavior of protein phases that we observe in droplets, he can arrest the growth of one polymer by using a second polymer, which forms this coating to minimize surface tension effects around the first polymer that's phase separated. And so this will, it's a very interesting work that's going on in terms of using the kinetics of phase separation to gain insight into the thermodynamics. So finally, I'll wrap up with outlook and challenges. So the basic Florey-Huggins framework is nice almost in a qualitative sense in that it allows us to understand things like multi-component interactions and so forth, but it cannot describe the phase behavior for more complex nucleic acid binding polypeptides. So these would be polypeptides that are enriched in charged or aromatic side chains, proteins that comprise ordered folded blocks concatenated with intrinsically disordered regions, and certainly there will be difficulties in describing multi-component systems where RNA is present. Okay, so this just represents from a nice review paper, the complexities involved in these multivalent interactions, things like charge-charge interactions, cation pi, et cetera. And so moving forward, folks will have to extend theory in order to account for these complications. And I think, okay, good. So I am not an expert or not have done much work in terms of simulation or modeling, but I wanted to include a couple of references in the tutorial from Rohit Papu and Clifford Brangwine, which shows some very nice work using coarse grain models. And so these will become more important as we start to include multivalent interactions in understanding the free energy landscapes. So just the last slide to show the complexities involved is there's a very nice paper which uses simulations to map the complete phase diagram of charge intrinsically disordered proteins. And so in this case, what they've done is they've just taken the charges, a minus charge and a plus charge, and jumbled them up. And they observed very different phase behavior based on just the location of these charges. And so it's easy to imagine the sophistication that would be involved in not only accounting for charges, but also hydrophobic interactions, as well as interactions with nucleic acids. So very quickly, I'd just like to acknowledge folks that I work with on these projects, Gabriel Lopez, Matt Lakin, Reed Selwyn, Andrew Shreve. I'd like to thank Michael Grunza for all the work he's done in coordinating between UNM and Max Planck. And I would like to thank my group, which is, there's a nice photograph of them. And I will be happy to take any questions.
Great. Thanks, Nick, for a great talk. We have a couple of questions here. I'll read them to you and you can reply. Sen Seneca asked, very interesting work on phase-separated, intrinsically disordered proteins. I was wondering whether work has been performed to correlate the presented sequence encodable assemblies to the compartmentalization of IDP-rich coacervates within cells. If so, which techniques or workflow can be used to study their phase behavior in more representative in vitro systems?
Yeah, so in this talk, I focused on, kind of very minimal repeats, but there's been some very nice work from Roy Parker from CU Boulder, which looks at various techniques to either recombinantly express native proteins or to observe their phase behavior within cells. And so I think that would be a really good place to start in terms of looking at experimental and synthesis techniques. Now, extending that into theory, the Papu's group from Washington has done some great work in terms of modeling these types of polymers. I forget the exact nomenclature, but it's kind of like beads on a string. And so they're beginning to account for order domains along with these disorder domains. And these are appear to be mapping on very nicely in terms of simulation.
Great. Sadaf Pashapur asked, how long can RNA be synthesized in the lab before it loses stability?
Oh, right. So if you'll notice in the work that I presented for my group, we're working with DNA, right? And the reason that we're working with DNA is for a card-carrying chemical engineer like myself, it is quite difficult to work with RNA. And so this is something that we're moving into, particularly as we want to create synthetic organelles that can regulate gene expression. So the short answer is I don't know the exact amount of time, but I know in general it's pretty short.
Great. Then the next question is Shika Dagar asked, does the properties, for example, permeability to the small molecule or RNA molecules of this membraneless compartment change upon undergoing temperature fluctuation cycles?
Yes, I think that's an important point, right? Is by controlling the solvent quality, which you can do through temperature, or you can do through, for example, chemical modification of your polymers, you essentially modulate the volume fraction that exists within the coacervates, right? So that's, as we talked about, that's what the binodal represents is, the intersection of the binodal represents the volume fraction in the coacervate phase. And so there are certain IDPs that, for example, can create rather large void fractions. And so I would envision that capturing nucleic acids as well as potentially processing them would be more amenable within coacervates that have these rather large volume fractions. And so, for example, as you increase the temperature of an ELP sample that's phase separated, in general, you increase the volume fraction within the coacervate, and so the void fraction is going to shrink. And so, I can envision that at some point it would no longer be effective at capturing nucleic acids.
Great. And we have one last question, again from Sen Seneca. Would it be possible to prepare a sequence encodable polypeptide chain that can stabilize these coacervates? Is there a limit to the chain length?
Oh, this is actually a very good point, and probably something I should have included. You can actually arrest the coarsening after spinodal decomposition by including a diblock polymer. And so the strategy is to create a protein where one block has the same sequence as the phase separating homopolymer, but you have a second block which does not undergo a phase transition. So these form essentially micelle structures. And so what these amphiphilic diblocks will do is they will coat the coacervate phase after spinodal decomposition. And by modulating the ratio of diblock to homopolymer, you can control the size of these formed coacervates from hundreds of nanometers up to microns. And so if you would like to see a bit more about that, this is in the Nature Chemistry paper that was within the presentation. Also, for any students, please feel free to send me an email if you would like any of these materials.
Great. Okay, so thank you, Nick. And I think that as we remain perfectly on time, we're going to switch over to Laura de Laporte. I'll go ahead and do the introduction. As a chemical engineer, Dr. de Laporte is an associate professor in the chemistry department at Aachen and research group leader at the DWI Leibniz Institute for Interactive Materials. She develops biohybrid and bio-inspired materials for advanced medical applications, such as replicating the complex structure of natural tissues. Professor de Laporte studied at the University of Ghent, Belgium, did her PhD with Professor Lonnie Shea at Northwestern, and studied regenerative hydrogels in Professor Jeffrey Hubble's group at the EPFL in Switzerland. From 2013 to 2018, Professor de Laporte led a junior group at the DWI Leibniz Institute and was awarded a starting grant from the European Research Council in 2015. In 2018, Professor de Laporte was one of 5 excellent researchers to receive funding from the Leibniz Program for Female Professorships. And with that, we are moving towards her talk on synthetic building blocks to grow functional tissues. Dr. de Laporte.
Thank you very much for the nice introduction. Thank you very much for everyone joining today. So my talk may be a little bit different than the talks you've seen. We are not really making protocells in the lab, but maybe the conference, like if we call it Syn tissue or something, then we would definitely also fit. So we use synthetic building blocks to grow functional tissue. And the reason why we are interested in this, I have to check now, yes, is one to develop new in vivo therapies to regenerate tissues after injury or after a certain disease, but also more and more there's an interest in these type of materials to make ex vivo tissue models. There's a very high demand by pharmaceutical companies in order to be able to make these models for drug testing, but also to better understand why certain diseases are occurring, like how specific stimuli are causing the onset of certain diseases. And so when we are designing these type of 3D constructs to grow tissues in a more artificial manner, there's always 3 types of stimuli that we have to try to mimic the extracellular matrix, which is normally the scaffold inside the body that is kind of guiding the cells how to behave and how to function. So these 3 types of stimuli are biochemical stimuli, mechanical stimuli, but also the architecture is very important. And this is also one of the focuses of our research group. So the type of materials we use are hydrogels. And the reason we're interested in hydrogels is because they're injectable. And also later we could then, for example, 3D print these materials to do bioprinting. And so in order to mimic these stimuli or to provide these stimuli to the cells, you can change the hydrogel properties by changing the architecture of the polymer precursor you use or the molecular weight or the concentration of the polymer. You can make the gels more stiff or more soft. You can make them swell more or less. But the polymer that we use in the lab is polyethylene glycol, and polyethylene glycol is completely bioinert. So it is FDA approved and also can be used, for clinical applications. But unfortunately, it doesn't have any biological function. But this is also something you could use for your benefit, because this way you can really control what you're adding into your hydrogel in a bottom-up manner and really see what each signal is doing to the tissue. So you can incorporate very short peptides, covalently link them to the hydrogel, or you can mix large ECM molecules inside your hydrogels. But you could also design specific ECM fragments, which have then specific domains from these large molecules and grow them, for example, produce them in E. coli and then couple them covalently to your hydrogels. On the other hand, these synthetic hydrogels have a very small mesh size, like a nanometer mesh size, and cells are like between 10 and 40 micrometer. So a cell would be completely squished in this matrix if it wouldn't degrade. So you can only use these type of materials if they're degradable. So you can, or you add hydrolysis group via ester groups, but you can also use MMP degradable cross-linkers, which are peptide domains that can be degraded by matrix metalloproteases, and matrix metalloproteases are enzymes produced by the cells. And this way, we now have a material that can degrade on cell demand. So not too quick, not too fast, but really as the cell needs space, the material makes space for the cells to grow. So one of the building blocks we used to make like these conventional hydrogels was using as a system that was developed actually quite many years ago by Jeffrey Hubble and Martin Erbar and Matthias Lüthold. And this is a synthetic fibrin gel. So what they did is they took a PEG polyethylene glycol precursor solution with a vinyl sulfone at the end, and then via a peptide which has a cysteine, so they have a functional tile, they can do a Michael type addition and they can click peptides to the arms of these precursor solutions. And on the other side of the peptides, they have the peptides that are a substrate for transglutaminase for factor 13, which is the same enzyme that is used to make the fibrin like a blood clot in your body if you hurt yourself. In addition to these reactive groups we need in order to cross-link the stars together, you can also include these MMP degradable domains so the cross-links can be degraded. So after you add the enzymes, the stars can link together. And now we want to make it bioactive. And in this case, the results I will show you are based on a fibronectin fragment we produced, which has two specific domains of fibronectin. One is a cell binding domain, so it binds to the integrins of multiple cells. But then the other domain is a growth factor binding domain, because the extracellular matrix in the body does not only attach cells, but it also binds a lot of different soluble signaling molecules. So by bringing these two domains together, you really bring the growth factors close to the cells and you get a synergistic effect. And so when we optimize these gels for different cell types and we couple the fragment inside, we could grow fibroblasts, or we could start forming these blood vessels if you have a co-culture. But we also grow these primary neurons. And these primary neurons we take from embryonic chick in eggs that we grow in the lab, and after 10 days we can take out these dorsal root ganglion, which are connecting the central nervous system with the peripheral nervous system, and then you have this cluster of nerve cells, but also Schwann cells and other supportive cells. And then you can put these clusters in your hydrogel and evaluate if the nerves can grow into your 3D hydrogels. And in this case, you see they grow very well, but they grow in all directions. So this is really like a radial growth. So this is a first start, but this still has a lot of challenges if you want to use this, for example, for in vivo therapies, because these materials are very isotropic and they don't have any alignment, and many tissues in our body, not just nerves, but also muscles or heart tissue, are more anisotropic and are not really isotropic. So we need to find a way to have an injectable material that will form an anisotropic structure after you inject it inside a soft tissue. On the other hand, these images I just showed you, the cells are then mixed inside the hydrogel before it cross-links, so when it's still a liquid, and the cells have nowhere to grow, so they produce the MMP, the material degrades and they can grow. But if you would inject a gel without cells in the body, then the cells have a much harder time to grow into your matrix because they are preferring to grow around the hydrogel. They have to somehow work too hard to infiltrate. So ideally, we should also move them towards like microscopic pores where you would have an injectable material that forms these larger pores. And based on the talk we just had, phase separation would be like also a potential approach you could use here. So another a problem here, but if you want to grow these tissue, is that you need these growth factors in order to direct the tissue, like how to form, for example, to mature blood vessels, and you need different growth factors at different times. So ideally, you want to have little drug delivery containers into your matrices where you can control the release kinetics at different points of these growth factors. And so for this, we make these hollow microgels via also water, oil, and water emulsion. In the oil phase, we have a polymer. I will put my laser pointer, a polymer solution which can then cross-link via UV, and depending on the shell thickness, the degradation rate, but also the mesh size, we can control the diffusion of the proteins that are loaded in the core, and you have a very high encapsulation efficiency into these materials. Besides the materials, cells, of course, can be very therapeutic. If people use therapeutic cells, one problem is still that a lot of these cells will like migrate away or will die. And so we also think that potentially putting these cells into our injectable building blocks, that we could protect these cells better from the immune system, but also keep them more local at the injury site and already provide them certain stimuli they may need to form functional tissue. So here we make these rod-shaped microgels where cells can spread inside. We also use co-culture. We did this together with Professor Akashi in Osaka University, where we use cardiomyocytes derived from induced pluripotent stem cells. And if we have a special cell coding, we can increase cell-cell interaction into these little mini beating hearts. And only with the cell coding, actually, we had better beating, more physiological beating than if we wouldn't have the coding. So let's go to our main focus in the group is spinal cord repair. The problem with spinal cord injury is that these patients usually don't have any therapy right now available in the clinic. So a lot of them are partially or completely paralyzed below the site of injury. And in 1980, we found out, and other researchers found out, that if you stimulate the central nerves with the right stimuli, that you can actually stimulate them to regrow again. But then people also realized if you don't provide any linear guidance for these nerves to grow across the injury site, they will just kind of grow in all directions, which is not ideal, of course, for such an oriented tissue. So people started making scaffolds with aligned channels or aligned fibers in order to guide nerves to grow across the injury site, also including myself during my PhD at Northwestern. I was making these multiple channel bridges. But then during my postdoc, I really wanted to see, can we also have an injectable material potentially, which would be low invasive if a patient comes in, that the surgeon will never cut a piece out of the spinal cord to implant a scaffold, but maybe they would inject something. And so after learning about hydrogels, then we came up with this idea to try to combine best of both worlds. And so with this, we created the Anisogel, which is a hybrid material which has these rod-shaped microgels that we produce, or short fibers. And inside we have super paramagnetic iron oxide nanoparticles. And so because of the magnetic nanoparticles, these microgels can align. And the second part of the solution is a polymer precursor solution, which will then cross-link, and it will keep the aligned microgels in place so that the structure would remain to guide nerves across the injury site. So now you could also remove the magnet because the rods will be completely fixed in the matrix. The way we make these rods, one way is to use a batch process, which is a print technique. So you put a polymer solution into these cavities, which you could choose whatever shape you would like. You need to make sure there's no film in between, that you have individual microgels. And then you can put the spines in the polymer solutions beforehand to make them magnetic. I just actually decided to add a few slides based on the previous talk. One problem we had is that if you fill these very small cavities, that a lot of the water, if you go to a very low polymer concentration because you want to make soft microgels, a lot of the water evaporates and we wouldn't have really nice microgels. So we decided to not use water as a solvent, but to use a non-reactive polymer. So a start pack with propylene oxide or a very small polyethylene glycol, which is still liquid, but just has the non-reactive OH groups. And when we did that, we saw that we got phase separation. So if we just make a gel with water, we get this kind of gel. If we use our non-reactive filler, we had these much larger pores than if we just would use water. So here you also see like our studs where you can see the larger pores. Interestingly, depending on the filler we use, whether we use like a very short polyethylene glycol, or we use a larger star which has 20% propylene oxide to still keep it a liquid to reduce the crystallinity, we have a completely different structure. We get this more interconnected structure, or we get this more globular. It's almost like binodal versus binodal decomposition here, although this may be a bit too far-fetched. But we also see that the mechanical properties and the swelling behavior of these gels are very different. So I don't have too much time to go into detail. I just wanted to show this also based on the previous talk that maybe this could be interesting. So in order to make the magnetic, we put these spions inside. And so here you see the distribution of the spions. Luckily, they interacted with our reactive phase, so they didn't wash out with our filler after we washed the filler out. And if we put it in a magnetic field, then these microgels orient in a very short time, within a minute. This video is now 5 times quicker than normal speed, but so they have to orient quite quickly because they have to orient before the surrounding gel crosslinks. And if the surrounding gel crosslinks, you get a construct like this, which we now call our anisogel. I mean, the black is a gel, which is keeping everything together, which is not stained. And if you mix cells inside, they will now grow in the alignment of these oriented rods. If you have a normal gel, then the cells will just spread in all directions. So we wanted to see if the cells are aligned because of our rods, they will also automatically produce their own extracellular matrix. Would the extracellular matrix they produce also be aligned? And so we did see that this was the case. These were fibroblasts, and the fibronectin is also aligned. So this kind of makes us think that this is a positive feedback cycle where now our microgels could disappear and then the natural system could take over and make their natural oriented extracellular matrix structure. So this was interesting to see, and we also realized that you don't want to overstimulate the cells, because if we were putting RGD onto these rods, then the cells actually reduce fibronectin a lot less. So we're kind of overstimulating the cells, and we're then missing out the chance for them to produce their own natural matrix. So we are trying to jumpstart the system with minimal amount of cues necessary. So this is also the case, of course, when we think about how many of these microgels do we put in. And so here you see the same dorsal root ganglion I showed you in the beginning, but now they grow really not radially anymore. They grow along the orientation of the anisogel. And if we go too low in concentration, then the nerves would grow in all directions again. So then we lose the orientation. You can also dissociate these nerves, these DRGs, into single neurons, and you can see this here. And we found that we can go down to 1 volume% of these microgels into the volume of our complete gel. And then they have like an average distance of 34 micrometer where the cells are growing in between. They don't have to be in contact with the microgels. The microgels are also completely inert. There's no cell adhesive molecules here. The cell adhesive molecules are in the black gel, which is around, which is keeping everything together. So then, of course, we were interested, and this looks a bit crowded, but I'll just talk you through it. We were interested in what is the ideal size? What is the ideal distance? Like, can we really minimize the amount of cues we have to add? Because this way we can also minimize the amount of spions we need, the amount of synthetic material we need to inject, so which would all be better for clinical use. So this was our standard condition, which was 5 micrometer width and 50 microliter length. So the first thing we did was to make them half as small with the same length, so keeping the length constant. We see we lose a little bit of the alignment, but this was still below our acceptable line here. But if we put them, of course, then closer together by adding a bit more microgels, we could further improve this. And we could then even reduce the length from 50 to 25. And so now if you look at the volume percentages, even we get very nice alignment here, but with much less volume percent. So we could really reduce the amount of synthetic material. And the good thing about the smaller microgels is that the total outgrowth of the nerves was much higher. So the rate of growth, not just the alignment, but the rate of growth was also much higher. And this we then realized must have to just do with the cross section of the microgels, and we saw that if they're very small, that the axons that are growing on are not really hindered. They can easily pass if they like hit one of these microgels. But if the cross section gets bigger, it starts hindering the nerves to grow. So now this is really like our optimal condition where we have like 2.5 micrometer with an aspect ratio of 10. So of course we need to test if this material works in vivo, and we just got our animal protocol approved. So we will now start this month, finally starting to plan all the animal work. This is a dead mouse that we got from the animal facility to just already test the injections, where you can see that also in vivo, if we bring the magnet close, we can, with a very easy system, align the anisogel into these mice. Of course, you don't see any nerves growing because these are dead animals. Hopefully more to show then next year. So as I mentioned, the directionality is one thing, but ideally we want to also have a surrounding matrix that easily lets cells to infiltrate into these materials. And so we may have to change the outside of the material into a more microporous hydrogel in the future. And so one more continuous way to make these rod-shaped microgels compared to the batch method is to use microfluidics, which I think you've already seen a lot of microfluidics during this symposium. So in this case, we want to have elongated drops because we cross-link on chip, and so you need a very quick cross-linking mechanism, otherwise they would snap back to a sphere after they leave the chip. And so this way you can produce a very high amount of microgels in a very short amount of time. And they're very soft also, they're very flexible, as you can see. But the problem with microfluidics is that this diameter is limited to 10 to 20 micrometer, the minimum, because of the pressures you need to push the fluid through. So these microgels are much have a higher width than the ones that we were actually using for the anisogel. So we wanted to find a solution for this and kind of overcome this limitation. And we thought, what if we make a jet with a polymer solution and not make these droplets? Could we now use an oscillating light to have like cross-linking, not cross-linking, cross-linking, do it alternating, so that the cross-linked parts would stay together after they come out and the non-cross-linked parts would just dissolve. So that was the idea. So first we wanted to see proof of principle. We have this hydrogel fiber just to show that it was cross-linking with a continuous light on. And then we started oscillating the light, and we got these very weird shaped paprika trumpet shaped kind of constructs, because if we were putting on the light locally, the jet was destabilizing, like slowing down the jet, and we were not able to separate them. So then we added, again, a non-reactive filler to stabilize the jets. And this time we saw these like peaks coming out, and these peaks, when we gathered the product in our beaker, we saw that this was actually what we wanted, that we had these very thin rod-shaped microgels that were now 10 times smaller than the chip diameter. So in this case, we used a chip that was 80 micrometer, and we could go down to 8 micrometer. We now are trying to do this with a smaller chip, like 20, and go to 2, but it's more challenging than we thought. So this is still ongoing. So here you can change the aspect ratio by changing the on light. If you have the on light, the on time longer, we have these longer strings, and if we have the light on for a very short amount of pulse, then we have these very short strings. And so here, our vision here is that we would not really use polymer precursors to make a hydrogel, but that we would use these preformed rod-shaped microgels as a precursor to make a hydrogel in order to get a more microporous structure. And so just again to show you these internal structures, because I think these are very important for diffusion and also later like actuation of these microgels. If we do the jet gelation, we have only a very tiny amount of light exposure to start the free radical polymerization, and we see that we have quite large pores, and these materials actually swell a lot. And so with the print technique, we had the phase separation, so the pores were all, the mesh size was also larger. But if we do the print not with a free radical polymerization, but with a step growth polymerization, again, using the non-reactive filler, we didn't get the phase separation. So it really also depends on which chemistry you use, which method you use, what kind of internal structure of these microgels that you're getting. So if we assemble these microgels together and we now couple RGD to them, we see that the cells can grow into these 3D microscopic structures. And we have like these large pores in between in which now blood vessels could grow in order to provide nutrients to the growing tissue, which is of course also important for long-term survival inside the body. So then the last part of my talk, I'm kind of switching gears a bit, even though this technology that I will show you now in a 2D system, the goal is to translate this to our 3D systems that I showed you before as well. But so in this case, of course, you need another polymer because we want to start having real actuation in our materials, like real movement into our materials. And for this, we use thermoresponsive polymers that can shrink above a certain LCST and they swell below the LCST, and we want to do this around 37 degrees for the cells. So we have a copolymer of polymepan and polyneum at 60-40 ratio in order to have a transition temperature of 37 degrees. And so here now with near infrared light, if we put gold nanorods into these hydrogels, we can locally heat up the gel because the gold will start heating up here because of the light. And then you can locally shrink the material. And if you do this now with oscillating light, you can do this very quickly up to 10 Hertz. So in this video here, this is like a holographic microscopic image we did in collaboration with this company in Switzerland. And so here we have 1 Hertz, and just to show you that also the amplitude of the force you can change by having an on time of, for example, 100 milliseconds or only 20 milliseconds, and you have a smaller amplitude. So different forces that you are exerting on the cells. And these gels were made with a certain pattern, 25 micrometer groove and 25 micrometer ridges that we then coated with fibronectin to grow the cells. So what we noticed if we actuate the cells, so from the 25 micrometer, there's like a 4 micrometer distance of actuation. This is 1 Hertz also. And if we do this for 24 hours, we see that after 8 hours, MRTFA starts shuttling into the nucleus. And MRTFA is a mechanosensitive protein which holds G-actin in the cell. And so it holds the G-actin, and when a cell wants to form more F-actin, it releases the G-actin, it goes to the nucleus, and more F-actin is being formed in the cytoskeleton. And so this observation was then consistent with the fact that we saw that these cells migrated less than on the region that was not actuated. So they were adhering like a baby afraid on a jumping castle. They were really adhering strongly to the gel. And this was depending on the frequency and the amplitude. If we did it at 10 Hertz, for example, we didn't see this effect anymore. And also at lower frequencies, we also didn't see it. So it was really at 1 Hertz that we saw this effect happening. In addition, we looked at the focal adhesions. We saw more focal adhesions. We also saw that the cells that were actuated were producing a lot more of their own extracellular matrix. So you see the fibronectin production here. Here we use a collagen coating, so we could stain for the fibronectin, and also this kind of aligned structures of these fibronectin molecules that the cells were producing. So this system, we now want to look more into like stem cells, also cardiomyocytes, muscle cells, see how this actuation can influence these different cell behaviors. So here you see, again, like an overview of what's happening. The cell is just not moving as much on these actuating gels. The MRTFA lets the G-actin go. You get more of this F-actin formation. You have more focal adhesions and more fibronectin production if you actuate these cells on these very soft gels. So it's much softer than, for example, PDMS, like these PDMS stretching devices. So with this, I come to the end of my talk. I have a short summary here to kind of sum up what I was hoping to convey to you today. So our aim is really to mimic the extracellular matrix using these smart polymeric biomaterials. And we always have to keep in mind that we want to have this bottom-up design for like biochemical, mechanical, and structural properties. We like to use these polymer precursor solutions because they can be injected and they can cross-link in vivo after you inject it, which is of course also a challenge because you have to pre-program everything in your material that it does what you want after you inject it. These magneto-responsive microgels or short fibers, which I couldn't show you today, they can align and direct cells in a unidirectional manner. Then if we would use these rod-shaped microgels to replace the polymer precursor solutions, we could potentially also have a more macroporous structure around it, so cells would infiltrate a lot quicker in vivo. Then, of course, we need the growth factor delivery inside our materials. So for this, we need to incorporate these hollow microgels that we are also now trying to make smaller, because the ones I showed you were made via glass capillary double emulsion, which they're quite large. These were 200 micrometers. So we are now also trying to translate this to PDMS chips. And then, of course, we could potentially work with other groups together and put very powerful cell transplantations into our microgels and combine this as a therapy. And then finally, we also aim in the future to combine these both parts of the talk, where we could start actuating these 3D materials as like an in vivo gym, for example, or like if we would bioprint these materials to see like how is certain actuation and movement during the maturation and the growth of this tissue, how much does this play a role? And could we also mimic maybe what kind of mechanical stimuli would cause certain diseases or certain processes which you may want to avoid, of course. So with this, I would like to, of course, most important slide, thank my group. I mean, I'm very happy to have a very creative and ambitious group that's willing to work a lot in the lab to make these dreams come to life. So I'm very thankful for them to do all the work. Of course, collaborators, because I truly believe that with many more brains together, we're all much, much smarter, can do more exciting research. And of course, funding, because without funding, we cannot do our job. And you, of course, for your attention. And I would be happy to answer any questions.
Great. And so we do have a number of questions here. Sen Seneca asked, incredible work. Is it a possibility that the reduction in cell migration is related to the organization of actin cytoskeleton above a certain threshold of actuation?
Yes, I mean, I can't really, I don't know. I mean, it could be. So this is where, of course, it would be, I wasn't, like I was introduced, I'm a chemical engineer. So of course, this is where I would love to work even closer with cell biologists and try to understand like what kind of cell pathways and molecular pathways are involved here. I mean, we all know how many reactions are included in the formation of a focal adhesion and an adhenosome. So that would be very interesting. But so yeah, there is something is happening also at a higher frequency. I'm not sure. We didn't look in more detail into these cellular pathways.
Great. Sadaf Pashapour asked, would using frequency higher than 10 hertz actuate MRTFA faster to the nucleus?
So higher than 10 hertz we weren't able to do yet. So 10 was the highest we could do. And we didn't see, so for one, we saw the MRTFA shuttling. For 10 hertz, we didn't see it anymore for the same amplitude. Of course, I do have to say, the system has its limits, right? Like you are creating heat by shining the light. And of course, you don't want to heat up the cells. So we, of course, have all the controls where we make sure it's not the heat that is causing an effect on the cell. But the amplitudes, for example, are limited in a way also, like how much heat we can put in, because we always want to stay between 36 and 39, like we don't really go over it. But if you want to have higher amplitudes, you need to put in a lot more light and a lot more heat. So this is something that we are still working on to kind of push these boundaries a bit. But so for now, we see actually that it's not shuttling to the nucleus at 10 hertz.
Okay. Andrew Halla asked, for the actuating gels, do other mechanosensitive proteins like YAP or lamin A also respond in a similar way to MRTFA?
Very good question. So we stained for YAP. I think it's also in the supplementary data of the Nature Communication paper. And we didn't see this, and the problem was that, so the YAP shuttling, the stiffness of our gel, the elastic modulus of our gel is actually exactly in that region where it's already, the YAP is already shuttling to the nucleus. And so we then try to make them softer, but if you then make them softer, the actuation also changes. So in the region that we were currently working, we didn't see the effect because it was already in the nucleus.
Okay.
On this gel, yeah.
Sen Seneca again asked, is there a correlation between the gel modulus and the folding behavior of important proteins like talin and vinculin, which we know are involved in focal adhesions? In other words, how does mechanosensation influence focal adhesion formation at the molecular level?
Yeah, this is again kind of to the first question. We would love to go more in detail. So we spent 3 years developing the system. And then seeing the observation with the MRTFA, again with the YAP, we didn't see it. And so all these questions to go more into like what is really happening inside the cells and which molecular pathways, we have to go more deeper into this. And so if people are interested in collaborating with us there or testing their specific cell types, we're definitely open for this. We did stain for vinculin and paxilline, that's in the paper. So that's, we saw the difference in focal adhesion.
Okay. Here's another question from Sen. Do you think you can use an on-off photo trigger with the nanoparticles to heat up the microgels and thereby be able to modulate cellular differentiation? I might assume that depending on the stiffness and gel modulus, things can change cellular differentiation, the cellular differentiating behavior of stem cells.
Yeah, I will do a little pre-announcement here, which I'm hesitant to say it because we saw something happening and then we have a hard time repeating it. But I'll just say it, but just keep it as a maybe. So we were putting mesenchymal stem cells on these actuating hydrogels, not microgels, just the hydrogels, but we could translate this to the microgels. And we saw that because of the actuation, it looks like we were having more bone differentiation. And that this was happening without using any differentiation factors in the media. So really just by the mechanical stimuli, it seemed like we could really push them into the bone differentiation. The problem with these mesenchymal stem cells is that they're coming from different donors and different patients. And there's such a large variability in these cells that a lot of these cells we get, for example, already seem to have a predetermined path that we have had a hard time influencing it. So we are now also working with another partner who's using induced pluripotent stem cells because we hope that there's less variability between the cells. So with one donor, it was very clear and we saw it, but then for the other donors, somehow it was more difficult to show the results. So this is very still preliminary, but we're working on that.
Great. And I have a slightly selfish question, both for you and for Nick before you, although Nick shouldn't chime in now. I really, really liked your anisotropic microgel objects. Those were cool. To what extent do you think that objects like that could be organized by nucleic acids on their exterior? Can you use hybridization as a rational tool to better organize sort of the gels and tissues that you're building?
I mean, sure. I mean, people have shown a lot of DNA like to use to cross-link. So of course you could use complementary DNA strands to assemble materials. There's also a question about price, of course. These are still quite large volumes that we're talking about. But importantly, for the alignment ones, there you don't want to link them together. You want to have enough space in between them that the cells have space to grow. What we would like to see also, because when a cell, and there we actually did see YAP shuttling. So we see that if you have more of these rods, we see more YAP shuttling inside the cells than if we have less of the rods. So this is in the biomaterial paper, I think. And so when the cell is in a matrix and it's pulling on this side or on this side, it gets a different feedback from the anisotropic mechanical properties, and it decides to grow in one cell in one direction, even if it's not in contact with a microgel. And so what we would like to see is what if we couple the microgels covalently to the matrix? Because in this case, they're just embedded. What if they're covalently or via DNA, like nucleic acids, bound to the surrounding gel? If a cell is now pulling, it must get a different mechanical feedback if this is connected. And this is something we haven't tried yet, but I'm pretty sure maybe we can even reduce the amount of rods if they're connected to the surrounding matrix, because maybe the force, how the forces are then dissipated, may be more efficient. But I don't know. That's just a hypothesis. But yeah.
Great. Okay, so thank you again, Dr. De Laporte. And now we, I think, are segueing to our keynote speaker, Drew Endy. I'll do the introduction for Drew. Drew is an amazing scientist. I should just say myself at the outset. He's a member of the bioengineering faculty at Stanford University and the founder of the BioBricks Foundation and president of that organization. His research teams really have initiated synthetic biology as a field, but pioneered amplifying genetic logic, rewritable DNA storage, reliably usable standard parts, and genome refactoring. Drew helped launch the new undergraduate majors in bioengineering at both MIT and Stanford. He co-founded the iGEM competition, many of us which know is a sort of global genetic area engineering Olympics that engages over 6,000 students annually. In 2013, the White House recognized Drew for his work on open source biotechnology. He recently received an honorary doctorate from the Technische Universiteit Delft. Drew served on the US National Science Advisory Board for Biosecurity and the Committee on Science, Technology, and Law. He currently serves on the World Health Organization's Smallpox Advisory Committee. Drew was a co-founder of Gen9, a DNA construction company, and he returned to serve as director of Gen9 during its recent acquisition. Drew worked briefly with the rapid evaluation team at Google X and also on the building project team for the Shiriam Center at Stanford. He is he's a founding co-director, of the NIST Stanford Joint Initiative on Metrology and Biology, but this is the one I like best. Esquire magazine recognized Drew as one of the 75 most influential people of the 21st century. So we're very pleased that Drew was able to find his way here, and he is going to give us an awesome keynote on upwelling, challenges arising from the bottom of Earth's life well.
Andy, thank you so much. Can you hear me? Just checking.
Yes.
It's been, thank you so much. And thank you everybody for your patience with me. I've always experienced the joy of meeting in person and the challenges of navigating the weather having to do with travel. The lesson I've had this morning is that the internet has weather as well. And a little bit more complicated than I expected to be able to join. But thank you so much, Andy, for that very kind introduction. It's good to see you and hear you. Kate behind the scenes, thank you for helping me navigate getting to here, and the headquarters for the meeting. Thank you for pulling us together, and let me go. So I think what I'll attempt is a screen share and talk about upwelling. Yesterday, I guess NASA and SpaceX had to postpone their launch of the 2 US astronauts up to the International Space Station. That was a exercise in coming out of a gravity well, a different type of upwelling, a physical upwelling, if you will. I've been thinking a lot in the context of building cells about a different type of well, what I think of now as a life well. And just like a gravity well constrains us in a position in space-time, I think of the life on Earth as a type of life well which constrains life from the perspective of the life that came before, the lineages, and also the practical constraints of needing to be able to reproduce and probably needing to be able to evolve on longer timescales. And so there are 3 constraints which limit life in a moment. And collectively, I think we're part of a time, and building synthetic cells in particular is at the forefront of the efforts of our time to climb up out of the Earth's life well. I want to start my talk, which will come in 3 parts, with a dream. because I think what we're doing is more, even, we know it to be hard, but I think it's even more hard than we realize. And we need more help than we might imagine. And I've been looking into the history, for example, of how much effort it took to go from the Rocketeers at the end of World War II to the Apollo program. And you might know quickly that's over 10 years to the start of Apollo. And I was surprised to learn from the history books that a lot of the people who wished to build rockets had to spend the better part of a decade teaching everybody to dream about going to space. I suspect that situation is more similar for us than we might know. So what, in Act 1 of my talk, if you will, dream could we share? Here's some dreams I have. They're not mine, but I am certainly smitten with them. Humanity provides for itself already, of course, but we do so in ways that are not always compatible with the rest of things around us. And so if you take these top two comments, enable humanity to provision for itself and stabilize and recover biodiversity, those seem like a pretty powerful combination, and very interesting factions could emerge in partnership. I've been spending a lot of time with colleagues learning about the conservation movement, also working with the International Union for the Conservation of Nature. One of the things I've learned is over my lifetime, the human population on Earth has doubled, and the natural biodiversity indexes have dropped by a factor of 2. Correlation for sure, but indications are that it's causal, that changes in land use and so on are leading to biodiversity decline. So what if that was different? What if we flipped that? What if there was a powerful partnership between the conservation biodiversity communities and the synthetic cell building communities, such that we could transition 10 billion people from living on Earth to living with Earth, an Earth Trek, if you will, not a Star Trek. To do that, we're going to have to figure out how to provision, as you'll see in a moment, joules, bits, and atoms, the physics of civilization at a planetary scale. How about some more parts of this dream? We're pretending that we're surprised by COVID-19. And of course, culturally and politically, in many respects, we are, and it's quite depressing in a professional sense. But just like if you were reading the newspapers before Sputnik was launched, you could have read in the New York Times up to a year before its launch that the Soviet Union was going to put that satellite up. And I think of COVID as a Sputnik-like thing, but what I mean by that is there's no reason to be surprised by disease, especially infectious disease. There's no strategic domain where the opening part of the strategy is let's wait to see what happened and then figure out our plan, and let's figure out how to react faster. That's the only place that seems to be true is epidemiology, which is bizarre. So let's have the dream of having infectious disease not be an operational challenge for civilization. And when we talk about civilization, we have to talk about the structure of that civilization. Is it a civilization of citizens? Is it a civilization of consumers? Is it a civilization of subjects to a king or queen? Is it a civilization of objects that are operating below an API controlled by algorithms? So I put these 4 up top because I think a lot of what we're motivated by and this amazing meeting highlights the last one, which is, wow, we really can learn a lot about life just by building stuff. Now you might be asking, what the heck does this have to do with synthetic cells? And I'm going to argue in a moment that all aspects of this dream are not just enabled by synthetic cells and a type of operational mastery of cells, cell construction, cell operation, cell modeling, but actually requires it. It's absolutely essential. I'm increasingly certain that we have to enable the operational mastery of cells in order to make these dreams come true. Let me try and put this dream in a context, which is very, very different than the context which dominates the tribes in the region I live, the tribes of Silicon Valley and the Moore's Law exponential. The problem I have with the dreams of the Moore's Law exponential is they just go off the chart, and it's not clear where we go, and it's not clear I want to be part of that. So here's a different dream. And I'll give you the nerd version of this dream. Not that long ago, well, of course, people, we require energy and stuff and knowledge of information. So I'm going to turn that into joules, bits, and atoms. You'll hear me use that phrase, joules, bits, and atoms. Not that long ago, our supplies of joules, bits, and atoms were sparse network supplies and usually limiting or just not available. Let's head to a near future, starting with the remaking of the facade of the Montparnasse Tower in Paris, which I'm looking forward to. Let's get to a near future, meaning within a human generation, where joules, bits, and atoms are provisioned at civilization scale in a way that's compatible with the rest of life on Earth. None of it's limiting. And it's going to be a new dynamic steady state. It's going to be a new basin of attraction. It's not going to be an exponential that shoots off into oblivion, the singularity. Who the heck knows what that is or if I want it. It's going to be disruptive still. It's going to be like going over that roller coaster where your tummy feels a little bit funny because it gets unweighted when it, ooh, right? It's like things are going to have to change. So want to admit to that. How would we know if we're doing it? Well, if we're talking about joules, bits, and atoms, we'd be able to make enough energy for civilization in a way that's sustainable. I'll come to that in a moment. We'd be able to use biology to grow or build whatever we need, whatever biology can grow or build anywhere. So instead of designed in California, made in China, or designed somewhere, made somewhere else, it's designed anywhere, grow or build anywhere, everywhere. And that's where the synthetic cell efforts come in as very foundational and essential. And we have to connect these capacities to human capacities. And I'll be biased. I wish for this to be led by Western democracies and to create a culture of citizenship representing the best of what a 21st century democracy can be. That requires everybody having the option of learning, but practically, I guess, 1 in 100 people are actively literate. So let me power through Act 1 with a few more details. A lot of times when you look at a system like a photovoltaic panel, you think about, as a consumer, how much money does it cost? How much electricity does it generate? What's the value of the electricity? And when do I get my money back? What's the return on investment? But think about it from a science perspective, from a physics perspective. How much energy does it take to make the panel? How long do I need to operate the panel to get my energy back? How long till I get my joules returned? And it turns out it depends on where the panel's made, of course. And you have to think about everything in the system, but this is one representation of it from 5 years ago. And it looks like with the scaling of the industry and careful attention to various efficiencies, the return on energy for photovoltaic solar generation went up to over 1, first off, so that's amazing. And let's say it's averaging 15 or 20. So what that means is if I have a return on energy of 20, over the lifetime of operating the panel, I get 20 times more energy back than it took to make the panel. That's never been true before for photovoltaic solar. And what that implies is that we're in the beginning of a transition to an electricity generation abundant civilization. I'm using photovoltaic solar, but you could pick your favorite generation source. That leads to some strange new openings, at least strange for me. So in California, we have problems with not enough water sometimes. And, of course, fresh water comes from solar power, evaporation off the oceans initially. How much energy would it take to run reverse osmosis and pumping to an average elevation of 1,000 meters such that you could provision fresh water to the entire landmass of the Earth? It's just a number, right? It's just an amount of energy. And the back of the envelopes I get to is about 20 terawatts of power. 20 terawatts is interesting as a point of reference because that's how much energy civilization's running on roughly already. Stated differently, if we doubled civilization's energy budget, we'd be able to run an artificial freshwater cycle. And that's in the limit where it never rains again. So that's just a mental exercise to warm you up to the idea that what would it look like to transition to a energy abundant civilization, energy generation abundant civilization. Now let's bring this back to biology. And maybe you've seen parts of this because it's fairly amazing. at least the stuff on the right, maybe you've seen, where organisms can be engineered to grow on things they don't normally grow on. In this case, a nice example of growth on formate. Now, formate's interesting because our friends in chemical catalysis and chemical engineering have been doing things like figuring out how to do electroreduction of carbon dioxide to formate, where you take an electricity and carbon dioxide and you get formate out. And when you do the napkin math on this, it looks like already a kilowatt hour of electricity gets you about a gram of biosynthesis if you were to put this together theoretically. And then when you talk to the chemical engineers who are thinking about improving the process efficiencies, they're thinking maybe a 30-fold scaling could be possible. A gram per kilowatt hour is already kind of interesting because it means 10 cents of electricity gives you a course of a medicine, but 30 grams becomes a lot more interesting from a biomanufacturing perspective. If this works at all, the macroscopic significance is we remove the cap on biomanufacturing that's currently gated by the amount of photosynthesis on Earth, 90 terawatts. That ceiling would appear to be removable, so to speak. So if I keep going on this, I can imagine two things. One, the personal biosynthesizer. It's a box. It takes an electricity, internet, which delivers DNA sequence or biocode, and air, which is a way of delivering nitrogen, carbon, and water. Closed loop engineering around this thing inside the box is going to be an interesting puzzle. DNA synthesis operating from enzymes in water without requiring phosphoramidites, but nucleotides, another puzzle. So lots needs to be made true. But if we could put it together, then basically people would have the option, not as a replacement to industrial biotechnology, which is what we have today primarily, not as a replacement to IB, but as a complement to that, people could do personal biosynthesis where they were. If only we could make all the things that are implied by an object like this true. What's the reality in the market? And I'm highlighting leading examples, not to pick on them, because I think they're best-in-class examples, at least coming out of the US at the moment. Two companies, one Zymergen and the other Ginkgo Bioworks. And it's just interesting to look at how they represent what they're doing on the website. Zymergen writes, our platform automation, machine learning, genomics to go faster as they're upgrading cells for useful purposes or designing cells anew for useful. And then they talk about their atheoretic approach. And the word atheoretic is so interesting to me because I have no idea what it means. It apparently means at first glance without theory, but even that seems to imply a theory. Now let's turn to Ginkgo. With the help of software-directed robots, 10 people at Ginkgo can equal what 100 people do somewhere else. Okay. So go robot power. At both places, basically what's happening is an Edisonian tinker and test methodology. Why? Because, well, we don't actually know how to design cells like we know how to design other things. We design cells through methodology that's very robust, but requires tinker and test. Now, why am I belaboring this? If you come back and think about a dream involving a personal biosynthesizer, I don't want to have to download onto my BioNet, onto my personal biosynthesizer, a research project. If I'm operating in a rural village in India, I'm not going to have the capacity to do research in the way I could do in Emeryville, California. I'm going to need to be able to download and operate content that works. I'm going to want maybe not these companies necessarily, but I'm going to want the experience that's the equivalent of, say, watching Mandalorian with my kids at night, or pick your favorite streamable entertainment. I'm not downloading a research project. In other words, I'm downloading content that's reliable that I can operate. So it raises the question, what's between now, our current reality, operating cells, and a future where we just download biocode and it works? Of course, we can do this in a limited sense for well-established genes and even viruses on occasion, but not at the scale of whole cells. And that's, again, where I think the synthetic cell movement and the synthetic cell operations really, really has to shine. Just one last part of this act, and after that, Andy, if I could get a time check with you to know how I'll gate the pacing of the two scientific vignettes I'd like to share from some unpublished work. This is a an argument of the time from 1813, Tom Jefferson to John Adams. And the gist of the argument here is how if you want to have standing as a citizen, you need to have the option of accessing the means of production so that we don't risk mutual oppression. Those are strange words to be using in this context, but they're interesting. If I have the option of doing something myself, of course, I'd rather buy it from somebody else who's better at doing it than I am. But if they're suddenly oppressing me through economic power or political power or other, by having the option that I can do it myself and others can do it themselves, perhaps for me, I'm much less likely to be oppressed. And if that's mutual, then we can have standing with relationships to our technology that are not only the standing ship of being a consumer, but the option of undergirding consumerism with citizenship. It begs the question, what are the means of production that are most relevant to 21st century citizenship? And what would it mean to give everybody the option of accessing those? And if you link it back to Joule's Bits and Atoms, I'm going to start with power generation, the wrangling of atoms, which is going to be biotechnology at scale, and knowledge, which we can handle separately.
So Drew, about 15 minutes, but we're pretty flexible.
Thank you, Andy. So that's the end of my dream. And the linking of the dream, if you will, to some of the hints of what synthetic cell engineering might need to deliver. I'm spending time on it because I really do believe all of us together have a duty to figure out how to engage with a shared dream well beyond the boundaries of this meeting, such that we have broad societal support for what we're doing. Okay, let me give two quick vignettes, both of which are unpublished. This first one I last presented a year and a half ago at the basic meeting at Delft. And so it's a little bit of an update on that. And then I'll get to a totally new thing. So a lot of us are interested in building things from the bottom up, so to speak. And one of the workhorses there for people learning is Pure, the purified components for cell-free expression. And Eric Way, an electrical engineering PhD student here at Stanford, got really interested in the question of, could Pure make itself? And let's just start with that, because if we could get Pure to make Pure, then other things are happening. Maybe we could put it all together. And you can think about this both quantitatively, like how many peptide bonds can Pure make, and can it make enough peptide bonds to reinstantiate Pure? And the answer is no, at least not out of the tubes. It's off by about a factor of 20, but that's okay. We can fix that later with energy. But it's also a qualitative question. Are the biochemical functions as represented in the purified proteins constituting Pure sufficient to remake those proteins with equivalent functionality? Are there some weird modifications in the E. coli that's used in the first place to make the Pure proteins that are missing from the Pure itself? And so it's really that part of the question, that qualitative question, that Eric's been exploring. And the way this has gone is we basically take Pure and we remove a component, and New England Biolab's been very gracious in helping us get access to alternate versions of the Pure kit without letting us build Pure ourselves. So the way this goes, for example, is if you have Pure, but it's lacking, say, the alanine tRNA synthetase, and then you have a reporter construct in there, do you get expression? Do you get fluorescence? The black curve in the top row shows with wild type Pure, if you will, you get nice expression. When you remove that synthetase, you get relatively little fluorescence. Then what you can do is you can take Pure, and you can use Pure to make the alanine synthetase from scratch, and then complement that in trans, if you will, putting that back into the depleted Pure to see if you can recover synthesis. That's what's shown in the bottom middle, where the blue curve pops up and recovers the activity indicated by the full native Pure, so to speak. So this is a nice example where you get good depletion when you remove the synthetase in this case, and nice recovery when you make it by Pure and then put it back in, implying that the componentry of Pure is sufficient to make this particular synthetase with equivalent function to however E. coli is making it. So the first thing Eric did was basically depletion experiments across all of Pure, just to see when you remove things, how often do you get diminished expression? And it turns out not as often as we hoped. And so the darkly shaded bars on the right show what happens when you remove one of the ingredients from Pure. Do you get reduced fluorescence in the absence of that ingredient? And for about a third of the things, not really. And that's problematic for answering this question because it means we don't have an assay basically to recover activity back from, if that makes sense. But the things in white off to the left, we think there's enough dynamic range that we can attempt to complement to see if it recovers function. And so this is just giving you a sense of a couple different examples, initiation factors on the bottom, the alanine synthetase on the top. If we look at the bottom two plots, for example, initiation factor on the left, histidine synthetase on the right, the initiation factor doesn't have sufficient depletion to give us a good assay. So we can't say anything about that by this assay. The histidine synthetase, it's not as clean as the alanine synthetase, but it is beyond the noise margins. And so we're confident in that red line being below the black line. And then when we supply synthetase made by Pure for histidine, it comes back up the blue line. And we're basically creating this block diagram where the top right color of the triangle, if you will, is how much depletion there is. The darker the color, the better. The darker blue color, the better. And then the bottom right triangle, the darker blue color, the better, how much recovery there is. So you see with this alanine synthetase, it's 2% activity when depleted, 108% activity when replenished. And then you see the colors are, let me just cut to the chase. So this is what Eric happily calls the periodic table, a way of keeping score regarding the answer to this question, can Pure make Pure? And for some of them, like the alanine synthetase, you see that blocks full in and it looks pretty good. For the ones in which it's magenta or red in the top left, and then it looks like a gray flag pattern to the right or stripe, we don't have dynamic range, so we can't say anything. And then the ones for which there's a top left triangle and an empty white triangle, we just got back into the labs this week. And so we're hoping to fill those in within the next few weeks and share the results with everybody. Working together, hopefully, to move beyond this and get better assays, but build upon central dogma bootstrapping capacities. Okay, so that's the update on Pure making Pure. I want to share something that's been in development as long, maybe 4 years now, but haven't talked about very much. And this is a real amazing and deep collaboration with a colleague in chemical engineering, Rosanna Zia. She's mostly a colloidal physicist, a fluid physicist. So her expertise is getting the physics right at the microscale for systems that are high volume fraction, meaning almost glass, very polydisperse, constrained by membranes, not just Brownian motion, but all the other hydrodynamic things that come to play when you're dealing with stuff like this. And we've been thinking about this in the context of building cells, in part because her passion is understanding the origins of life, going all the way back to Brown observing pollen grains. And my interest as an engineer is, well, I want to be able to construct cells. And we're puzzled by how many things we still don't know, essential genes of unknown function, or in this movie here, looking at protein synthesis, where we have the miracle, if you will, of knowledge-enabled structural biology. Note the black background. How are those ternary complexes bringing the amino acids actually arriving? And is it really the case that by some miracle of transport, before the RNA is decoded, the exactly correct ternary complex out of the 40 or so appears? Because that's what's shown in this movie. So how's that work? And so we've been playing with, not playing, but we've been working carefully with simulation tools, starting with Smolden, and then some of the other colloidal physics tools that are more rigorous from a physics perspective, and trying to find a way of faithfully representing colloidal hydrodynamics and reaction in a modeling platform. One way I think about this, and Andy, if you remember me long ago, before synthetic biology, my point of entry into biology as an engineer was modeling biology for and with the biologists, before it was called systems biology, what's shown on the right over here. And that's now well established. But I kind of ran away from systems biology and into synthetic biology because the physics engine of systems biology was like, it kind of doesn't exist. We abstract away space, for example, oftentimes when we do whole cell models. On the left, of course, we have very well established physics-enabled atoms to molecule structural biology. And so what Rosanna and I and our teams are exploring is what would it mean to fill in the space in between. You could call it colloidal biology or whatnot. But it's the domain that links the molecular scale down up to the cellular behavior scale. So we've been looking at protein synthesis, just to connect it back to Pure, as a model system for learning how to build these types of representations and to inform the cell building community how we might think about what functions need to be there beyond what we're already thinking about from a genetic and biochemical sense, what physical functions need to be there. And so what's shown here is a voxel representing the componentry needed to implement protein synthesis in cytoplasm. In this case, we're using E. coli as a model, and we're tracking abundance and volume such that we can get volume fractions of the different molecular components. And we can link it to things like physiology and growth rate and environment. For example, it turns out that as E. coli grows faster, the volume required to contain all the elements needed to synthesize proteins drops by about a factor of 3, meaning it also gets denser. So the volume fraction goes up quite a lot from 0.13 to 0.42, we're estimating. This is going to lead to very, very interesting differences in transport phenomena. And then the transport phenomena, when you go back and think about how does protein synthesis work, that's going to relate to the puzzles of the reaction chemistry happening at the ribosome. So just to calibrate you to what's coming in some detail, drawing from the literature, beautiful physiology work from Bremer and Dennis and others, we're estimating this is how fast the individual ribosome elongation time is in bacteria as a function of growth rate. So each peptide bond is going to be at a slow growth rate happening at about 80 milliseconds and say 50 milliseconds at the fastest growth rates. And this is actually a pretty interesting phenomena I wasn't too familiar with. I'd always just had this ballpark number of 17 amino acids a second or something like that. But it looks like the actual microscopic peptide bond rate or elongation rate for ribosomes is itself changing with growth rate. So the question we're using to motivate the tool development here is practically, well, would Brownian diffusion alone be sufficient to supply an elongating ribosome with ternary complexes? And we build some models for how to keep track of the time spent moving around, time spent mismatching, time spent trying to find a match. And then we build out voxels with different types of representations. The movie associated with the Nobel Prize from 5 or 6 years ago, that's just a black void of space in the background, that's showing instant perfect transport. And that's really the plots on the bottom left here. Single ribosome, cognate tRNA magically gets there. And when you look at it from that perspective, you see the time goes mostly into chemical reaction latency, the 42 milliseconds, and the transport latency, 0.08 milliseconds, almost nothing. But as soon as you start filling that voxel with real numbers of things, the transport latency goes way, way up and actually is about 5-fold greater in the physiological case compared to the chemical latency. It turns out when you look at this, there's really interesting puzzles and trade-offs. For example, as the cells grow faster and the density of the translation voxel increases, the volume fraction goes up. If you just look at plot A here, the things get closer together. So you would think that's going to be good for transport. And so the squares plot starting at the peak in the top left and going down, that's indicating the distance between the ternary complex and the nearest ribosome, starting at 25 nanometers at low growth rates and going down to 7 nanometers at high growth rates. So that was, oh, we're going to go faster. That explains the acceleration of the microscopic elongation rate. But then look at that triangle plot that's going up from left to right. As things get denser, the viscosity of the system goes up, which means motion will be hindered. And it looks like we're getting a 2.5-fold increase in viscosity. So there's these trade-offs when you think about the physics of composing molecules into operating cellular systems. For time, I won't belabor the bottom plot. But to cut to the chase, when we use Brownian motion alone to try and answer the question, well, can we get the ternary complexes there quickly enough? The green plot at the bottom of this new graph on the right, that's what the reported experimental rates of translation elongation are, milliseconds spent per making a peptide bond. When we run our best models that aren't fit, but are just derived from first principles, we're getting estimates of 2 times, 3 times too slow. Meaning Brownian motion by itself isn't going to be the only thing enabling the ternary complex to get there. That's our prediction at this point. And of course, it begs the question, what could be missing? And could we use this way of thinking, this colloidal powered bridge between structural biology and systems biology to find missing elements needed for cellular function before we need them, right? So I'm trying to position this to the cell building community is, just like the genome minimization efforts get us to essential genes of unknown function, the bottom-up cellular construction efforts, we can anticipate they're gonna get to a point where we've put everything essential in there and it's not working. Those are called the essential functions that are unknown. And so could we use tools like this that get better physics engines for representing whole cell behavior to find those gaps ahead of time? What the top plot's showing here is we think we have some ideas in this case for how it's working. Pre-tiling weekly of ternary complexes on the RNA perhaps, or preloading on the L7, L12 part of the ribosome to accelerate some of the transport issues. So preloading and pre-sorting mechanics, when we incorporate those into the models shown on the bottom right, just to cut to the chase, looks like we can recover the experimentally reported translation elongation rates. Again, this is a specific example, but meant to be a foray into building out a representational framework, a way of modeling entire cells that allow us to anticipate how to compose and operate cells as physical objects, not only genetic and biochemical objects. So let me close with this, a bookend to the opening. This is a little essay I wrote for Art Forum, which is looking at biology. They're a modern art magazine. I'll just play with the opening lines here. We are the well dwellers wallowing in our well bottoms. Our most familiar well manifests via the gravitational force keeping us physically here, a gravity well that we can escape by rocketing up, up, and away, escape velocity. There's a second well largely unseen, meaning we haven't widely recognized it, manifests via the life preceding us, defining us, and burdening us in the now as corporeal form and function, a life well that we might eventually escape by transcending terrestrial lineage, life liberation. Upwelling within our life well, this vignette ends, should wonderfully renew our well itself. So with that, I'll stop. Andy, thank you, and thank everybody for your patience, questions, or discussion.
Drew, thank you as always for an incredibly insightful talk and a great forward look. I asked questions from the audience to you, and Ludo Schoenmakers asked, thank you for a great talk. I feel sympathetic towards your dreams, and I have a somewhat political question about the relationship between the first two. Bringing together the biodiversity crowd and the synthetic biology crowd might be quite a challenge, mainly due to considerations of naturalness and complexity generally prevalent in both groups. Nature is sometimes thought to be too intricate, hence too difficult and dangerous to control. Yet that kind of control seems to be the only hope to realize your dreams. Do you see the issue of this difference in perspectives? And if you do, how would you try to overcome it?
Ludo, thank you so much for that question. And of course, you're absolutely correct. I'm heartened by the leadership within the conservation biology and biodiversity communities who are exploring these possibilities, it's very, very controversial. It's controversial for the reasons that you mentioned. It's also controversial operationally because the cultural controversy can lead to an economic controversy, meaning it threatens the business models at times of fundraising. But here's the deal. The data for biodiversity aren't great. Things are going extinct. The posture of the biodiversity community strategically for 2 human generations now has been reactive, and let's fight things going extinct. And if you take a frank step back and look, that strategy is failing. And it doesn't look like it's going to stop failing. So that creates quite a crisis, not just the crisis of extinction, but the crisis of culture. I was at the headquarters for the International Union for the Conservation of Nature. I didn't know very much about this organization until a few years ago, so maybe you don't know about it at all. It's an organization of organizations representing nature. It maintains the red list, tracking extinction events and threatened extinction events globally. Their headquarters east of Geneva, you can actually see the card catalog from decade after decade after decade after decade after decade of tracking of extinction events and what's going on. And of course, you can get it online now, but there's something quite overwhelming viscerally when you pull out a card catalog and you read an entry about a plant that no longer exists, or an animal that no longer exists, or an insect that no longer exists, and you recognize that somebody in 1950 or 1960 had to type that up in capital letters on that card and say extinct, and then file it away in the extinction cabinet. So the puzzle then becomes, do we build a bridge and can we get there? I would point anybody who wants to look into the sausage making at what the IUCN itself is facilitating, a working group on synthetic biology looking to bring guidance to the conservation community through their Congress about what to do about biotechnology and synthetic biology. Everything from the direct impacts, like do you release a gene drive or not, to the indirect impacts. How does biosynthesis impact land use? And how does that then change habitat? One last comment about this, and then Andy, I'll go back into pause mode. The notion of the human and the notion of the natural are constructions. These are constructions derived from thinking 400 years ago in Europe. The human as sacred, everything else is an object of study. I think, therefore I am. These ideas contributed significantly to group fitness at the time and still do. But this idea that the human remains sacred and therefore separate from nature, note that that is an invention of both the human and nature in a way that no matter how much we love nature, we're always going to love the human more. And there's a debt accruing because of that. And so there's a very profound, almost philosophical, cultural question around revisiting some of the products that shipped in the 1600s. And can we come up with something that brings humanity and nature back together? My favorite wordplay is human and earth making a hearth, but that doesn't solve the problem.
Great. Ravi Ram asked, hi, Drew, although energy abundance from photovoltaics is great, it's not been limiting us from doing what you mentioned. Civilizationally, we have been getting energy rich, parentheses, EROI of oil is incredibly high, and we've basically been getting it for free. So what's really limiting such developments? Not energy, not tech. We don't need CRISPR for this. Enzymatic DNA synthesis should have been already here. So why now?
Ravi, thank you for such an insightful question. Of course, I'll say I don't know, but I'll give you my best speculation. Let's start with the following. Technology is not technology. It depends on the form of the technology. So a computer is not a computer. The personal computer is not really a technology revolution. It's a cultural revolution. It complements the industrial computer because it changes who can access and practice the technology where to what purpose. More close to what we know in this meeting, a DNA sequencer is not a DNA sequencer, right? So the sequencers we got most first were the sequencers optimized for the public scale government and corporate human genome sequencing efforts. And so they optimize the technology on throughput, cost, and centralization ultimately. Now that's complemented much, much later with the nanopore sequencers, which are USB peripherals. So now in your question, you bring up synthesis. Enzymatic synthesis should already be here. Well, of course, it's here in prototype form already, but it's not here in practical form in any sense. And I would submit for consideration that it mostly has to do with the structuring of the landscape within which the technology gets developed. What is our dream? What is the form of the technology we wish for? And if we construct that in the correct ways, we'll get those forms of the technology. And it just hasn't happened yet. There's other things to say, that are powerful ambiguities, right? We've got 2 centuries of fossil-powered industrialization and centralization, which is massively beneficial. And how can we best complement that with network-enabled distributed manufacturing? Unknown and daunting to think about, going up against that. So I think it really does start with the dream, though. And so depending on what form of the technology you wish for, if you can get that dream out there and share that with others, then there's a chance it will come true.
Great. Zakir Tanimov asked, great talk, Drew, as always, very inspiring. I have 2 questions on Pure. 1, what may be the reason behind such low depletion levels with respect to amino-acyl tRNA synthetases? Contamination, non-cognate translation, cross-tRNA amino isolation? Go ahead and answer that one first, then I'll do the next one.
Perfect. Like totally perfect question. No idea. All possible. Would love to know.
Right. Yeah. Also, regarding your second scientific endeavor, while preloading mechanisms have been showed, what are the mechanism on pre-sorting?
Yeah, so 2 to think about. So you do get to play with a small number of hydrogen bonds between the anticodon loop and the mRNA outside of the ribosome. Question is whether or not you can get any biasing in space due to that. Other thing I've been thinking about is in prokaryotes, at least, as you well know, you get multiple ribosomes following each other. And so could you get a reuse, if you will, of a recharged ternary complex coming off the preceding ribosome? That by itself gives a little bit of a boost when you think about the average number of peptides made per transcript before RNA degradation, but it's not sufficient to recover the rates. So it'd have to be something else too.
Great. Mahesh Vibhut asked, thank you for the inspiring talk. In your simulations of diffusive transport of ternary complex as a function of volume fraction, it is interesting to see that Brownian motion alone does not completely account for translational efficiency. However, in your simulation, did you take into account the fact that viscosity of the cytoplasm is a function of metabolic activity in a particle size dependent manner?
Thanks for the question, Mahesh. Only in the sense that we're accounting for that through the reported differences in cell composition, cytoplasmic composition, due to changes in growth rate. So we're really, the variable we were sweeping through, because we had great data from Bremer and Dennis and others, was cell composition changes due to differences in growth rate specifically. And so only through that variable and not through any other variable, which would also be fun to look at. We just haven't done it.
Yeah. And so now I'm selfishly going to ask a question. It's been way too long since we've bounced brains. As you well know, I'm somewhat to the left of the military-industrial complex, but well to the right of you. Jefferson never envisioned the possibility of a cranky neighbor synthesizing something that would take out Virginia's tobacco crop. Why is your dream not everyone else's nightmare?
Yeah. So thank you, Andy. And that's the right question. So I'm going to come at that 2 ways. The first is to talk about speech and how we govern speech, because I think at the end of the day, what we're talking about in synthetic biology and synthetic cell construction is a form of speech. It's the expression of intention into a substrate, and the DNA synthesizer is a printer, is a printing press. Just happens to be a printing press for expressing human intention in living matter. So we govern speech in the United States. I'm not going to talk about the marketing of how we govern it, free speech, because that's not actually true. We govern it through two Supreme Court cases primarily, Jacobellis versus Ohio and Brandenburg versus Ohio. One having to do with obscenity and the other having to do with lawlessness or law and order. So is the performance of this French play obscene, extreme pornography? And Potter Stewart, in his ruling, very famous ruling, has the phrase, I choose not to define extreme pornography here, but I know it when I see it, and this isn't it. What he's doing through that ruling is basically allowing local environments to contextualize an evaluation of whether or not something is obscene or not. So we could perform a French play perhaps on this conference, but if we did it at the child care center, we'd be arrested because the context makes it obscene there. Now, how about law and order? So by your speech, do you intend to incite lawlessness? And are you likely to succeed? If this had to do with a Klan march, if both conditions are met, then it's not protected speech. You can't speak it. That's the Brandenburg case. So that's been pretty good because it's scaled governance for obscenity and law and order, including safety and security for speech. Now, before we get to biology, we can recognize that there's a big problem arising for that framework in general, having to do with the internet. Because suddenly the recording of a terror attack in New Zealand on a mosque gets rebroadcast everywhere that has access to internet, which I no longer take for granted apparently today. And so the containerism of local context that made these rulings work really well for speech governance seems to have basically gone away due to networking, right? Decoupling information from location and recoupling it. And nobody has quite solved that problem in general, in the general case, never mind for the bio case. So now we turn to the bio case. And I guess where I land on this, Andy, is we're going to have to place a bet. So the bet is, do we think we'll be better off with nature as it's playing out and industrial biotechnology as we have it and things leaking around the margins, which gets us to today and whatever comes after today? And I'm already annoyed enough that that's an understatement with enveloped RNA viruses, right? Like, why do I have to go somewhere to get an antibody test? Like, why can't I just have myself tell me if I have an infection? Okay, and then you well know, right, some of the prescient ideas, not without controversy, from colleagues like Roger Brent. It's like, well, we're simply going to change who we are and our environment such that we have prepositioned defenses against most of the major threat vectors. And there's some actually pretty interesting ways of doing that within the context of the human that don't require germline modification. So maybe we should revisit that.
Yeah.
But anyway, so I start with that. And then the bet here is, do you like the status quo? And I'm sure I don't like the status quo. I don't know where you sit on the status quo, but I think COVID is definitely attacking the status quo. Or can we take a shot at something that, yeah, my neighbor might be attacking my tobacco farm, but it operationally doesn't matter because I can see it happening and I can work around it and I can have them arrested, right? Because I can do all those things needed for governance to work.
Perfect. We are totally set up for a future joint TED Talk. As it stands, though, we're going to thank Drew again, who was awesome as always, and segue to Kate Adamala. And so here, let me do Kate's brief introduction. Kate is an assistant professor in the College of Biological Sciences at the University of Minnesota. Her research aims at understanding chemical principles of biology using artificial cells to create new tools for bioengineering, drug development, and basic research. Kate received an MSc in chemistry from the University of Warsaw, studying synthetic organic chemistry. In grad school, she worked with Professor Pier Luigi Luisi from University Roma and Jack Shostak from Harvard University, two incredible giants, and she follows in their footsteps. She studied RNA biophysics, small peptide catalysis, and liposome dynamics in an effort to build a chemical system capable of Darwinian evolution. Kate's postdoctoral work in Ed Boyden's synthetic neurobiology group at MIT focused on developing novel methods for multiplex control and the readout of mammalian cells. And perhaps appropriately, given the conversation we just had, Kate's going to give a program highlight on a democratization of synthetic cells called Build-A-Cell. And with that, I turn it over to Kate.
Thank you, Andy. I will share my screen, and so it's my pleasure today to give a really brief highlight of this amazing initiative that I'm honored to be part of. We call ourselves Build-A-Cell, open collaboration supporting the science and engineering of building synthetic cells. Build-A-Cell is not a formal organization in any way. We're a volunteering community that unites people that have this shared goal of trying to build a synthetic cell without defining a synthetic cell and without defining any rules other than be civil to each other, share data. Build-A-Cell started a few years ago as a completely kind of voluntary grassroots organization. Recently, we managed to get some funding. We managed to fund people to travel to meetings to unite the community. And one of the biggest achievements of the Build-A-Cell community so far is that we managed to self-reproduce. Recently, our South American collaborators started I don't know how to call it, chapter, a Build-A-Cell South America within the bigger Build-A-Cell community, expanding kind of our activities and creating a local venue for people that couldn't travel to our biannual meetings as much as we would like them to. It's hard to define what a Build-A-Cell is, and it's hard to define what our goals are, other than that we do not want to restrict anyone's research. We want to facilitate research without putting constraints other than common sense and safety. So in that way, we do not define what a life is. For an organization that works on facilitating building life, you would think we have a definition of what we're trying to build, except we don't, because there will be more than one type of a synthetic cell. As Marilyn said yesterday, the work will never actually be done. So Build-A-Cell does not define what exactly a synthetic cell is. We're just trying to build an infrastructure, build a framework, so people interested in any kind of synthetic cell engineering can work together and figure out among themselves what is the particular goal of that group of people. So Build-A-Cell as a community has more than one goal, way more than one. We want to facilitate building cells, but we don't want to limit anyone, and we don't want to restrict anyone. The biggest thing we're focusing on is coordinating research efforts, creating kind of abilities for people to share unpublished data, to optimize reproducibility of protocols, so we can work as a community. We can share protocols that we know work between different labs, and we can share reagents. That's one of the biggest kind of goals of our most active working group, is to try to figure out how can people physically truly work together despite being geographically separated. We're also focusing on outreach and education, building cells, as Drew mentioned, some aspects of it. Building cells is a goal that can be misunderstood. People can look at it and say, we're making something crazy, we're making something risky, and that's not true. And as a community, we're trying to focus on explaining what we're doing and why what we're doing is good and not, and how we're taking the risks into account, how we're protecting ourselves, how we're protecting the public. We're also trying to influence science policy. We would love to see our field receive more research funding. We would like to be able to facilitate collaborations with other fields of research to kind of deploy synthetic cells as tools for other fields and use tools developed in other fields for building cells, kind of building those connections between different areas of science and building kind of a funding infrastructure around this whole goal of engineering biology from scratch. We, especially recently, that became really relevant. We're focusing on biocontainment. We believe in think before you make. We have seen what happens if a novel organism goes around the world. We definitely are not there. As you all know, synthetic cell research is not and will likely never produce actual pathogens, but this is something we as a community are aware of, and we're thinking about it. And I'll go over the details a little more, introducing working groups. The most important thing about Build a Cell is that it's the organization that combines efforts of a lot of people from a lot of different backgrounds. We have a lot of awesome PIs that participate in one way or another, but I really want to emphasize that none of it would happen without the steering group. The whole initiative was started by Drew Endy, Richard Murray, and John Glass during one meeting at Caltech a few years ago. And now with Elizabeth Strykalski, Lynn Rothschild, and Vincent Neurath, this is the steering group of Build a Cell. These are the people that do most of the kind of a groundwork, the boring work of getting funding, trying to coordinate everything, trying to put the community together, obviously with help from a lot of PIs within the Build a Cell. And the biggest activity and kind of a one point of focus for our community are in-person workshops. We're meeting every 6 months. We've been meeting every 6 months since workshop 1. And during those workshops, we actually try to work. Our workshops are notorious for not having talks. We don't really spend time listening to one person talking for half an hour. What we do is we actually get together, sit down around the table and work. So working groups are kind of the thing that makes and defines a Build a Cell. Whichever working groups are active at any given time, that is what Build a Cell is doing at any given time. We emphasize trying to have tangible outputs from our working groups, things like protocols, papers, lists of priorities, lists of possible roadblocks, and everything that can help us integrate the community. Science Build a Cell is not a single research institution. We just want to kind of unify the community and define goals that the community finds interesting and important. We rely on the input from the working groups and rely on the input from people that make those working groups. The workshops had been happening since 2017. That was the first founding Build a Cell workshop. Right now, the workshop always has been in the US, all around the country. We're trying to move the workshops around so we can have people participate from different, without increasing cost of travel for everyone. And we're trying to kind of bring the workshop close to every local community. Next workshop was planned for this summer at NIST headquarters. Right now, it most likely will be a virtual workshop, and we will reschedule the next one. So this will be the first time when the Build a Cell community will not meet in person since we started. The thing that drives the Build a Cell, both workshops and all our other activities, are tangible outputs that we're trying to produce. So we're trying not to just sit around and talk about how wonderful it would be to build a cell. We're actually discussing specific steps we can take to get there. And all our data, all our outputs are open source, available online for everyone, not just members of the community. And one of the examples of one of the outputs that we finished is a cross-referenced, tested in multiple labs protocol for making liposomes that express proteins. So that kind of a basic chassis of a synthetic cell. Any of you who ever tried work like that, you're aware of how many problems with reproducibility of liposome protocols we have. And that was one of the first things that Build a Cell tackled, is let's create a protocol that then different labs will test and validate and annotate. So then we can have a protocol and say, here, take this, and with this protocol, you should be able to do this experiment without anyone actually having to teach it to you hands-on. And this seems to be working. We're working on right now on protocols for other other procedures essential for building what most of the community agrees is a synthetic cell. And all the work, all the protocols, all the annotations, everything is available to the public. So you can look at the history of making that protocol. You can see how we got there. And if you have any comments and annotations, you can go in and add your kind of your input. We have received NSF funding last year. Thank you very much. That gives us 5 years of support to continue our activities, continue meeting in person, hopefully, starting again this winter, maybe. The biggest kind of part of Build a Cell work, the working groups, are open to everyone. Even if your lab is not a member of Build a Cell, you're always welcome to join a working group at any time. And here I would like to give you highlights of a few of the most active, currently most active working groups. One of my favorite is the Life OS Roadmap. It's a group of people that are trying to write an operating system for life, defining all the major needs that will have to be met in order to build a synthetic cell. That gets awfully close to trying to define what life is and what a synthetic cell is. I don't actively participate in that working group, but they are looking for new members. You can go to our website. There on Build a Cell website, there's a list of working groups and contact info for people that you should contact if you want to join any group. Again, you don't have to be a member. Your lab doesn't have to be a member of Build a Cell. If you're a grad student or a postdoc or a researcher, you can join the work anytime you want. Another pretty active working group is an in silico cell group. Drew discussed similar work a little bit. The in silico cell group is trying to build a framework for modeling and simulating synthetic cells. They're also looking actively for new members. A very recent but pretty active working group that we started in light of the kind of a biosafety problems that the world is facing right now is an ethics and biosafety group. We're talking about not just ethics of building life, but also how to do it safely. Someone pointed out that building life from scratch is the ultimate gain of function research. You're taking something that's not alive and you're trying to make it alive. So this working group, again, is looking for new members, and we're trying to discuss that problem from all the angles, from the ethics to all the way to trying to balance safety limitations with keeping the research open and moving fast. Other active groups are the Interlab group. This is the group that produced the liposome protocol that I showed you a couple slides ago. And this is the group that's right now actively working on shipping reagents between different labs and cross-testing different protocols. Pure That Makes Pure is a group that's led by Drew's student, the student whose work Drew showed on his slides, trying to figure out how to make a self-replicating translation system. A communication group is a group that attempts to improve the slide deck I'm showing you right now. This basically the slide deck I'm using right here is something that I made on my own, and it's not as pretty and not as good as it could be. We decided we would like to have kind of a ways to communicate goals and give an elevator pitch of the Build a Cell, and that group is working on making that free shareable slide deck about what is engineering cells, what is Build a Cell. One of my favorite groups is the Essential Gene group, the group that is trying to figure out what are the genes essential for building a synthetic cell. And obviously that will lead at some point to defining what a synthetic cell is as well. They are trying to figure out how to get around that right now. Build a Cell is running a virtual seminar series. We meet on Monday at 4 p.m. GMT. And we have speakers. We started as a journal club. Now we're just a seminar series with people talking about the research. If you have something you want to share with the Build a Cell community, please sign up for the seminar, or please shoot me an email and we can find a time spot. And just to finish, Drew did a really good job describing what the community is about and how we should move forward. This is kind of my poorer attempt at recapitulating that. What Build a Cell wants to do is to integrate the research community. So we basically want to go from this, which is the current funding and collaboration situation where people are clustered geographically around their own research institutions and mostly working within small teams that are kind of defined by either funding structure or their academic affiliation structure. What we would love to do is to have a community that works regardless of the geographical and institutional boundaries, a community that works based on their shared interests, where people interested in a specific aspect of cell engineering can work together and can get funding together, regardless of which country, which continent they're on. So we would basically like to have a community that's working together towards a specific goal, regardless of kind of institutional and organizational boundaries, the typical boundaries that have been organizing research for quite a while. And the reason for doing that is because building a life cell from scratch is probably one of the biggest challenges in biology right now. And no single university, no single nation should own rights, specific rights to this technology of building cells from scratch. And to do that, we need to build an integrated international community. We're having excellent examples of how the European community is integrating across the country boundaries. Now we would like to extend that, and we would like to connect everyone who works on building synthetic cells and give people kind of framework to work together and to develop shared strategies without having one voice kind of speak over everybody else, without one strategy dominating the field. So for example, if you think a synthetic cell is a liposome that makes proteins, great, go find a group of people and work on it. But if someone else thinks a synthetic cell is an oil droplet, fine, go work on that too. We're basically trying to build a lineage agnostic framework for people to work together without limiting what can they work on. And thank you very much for giving me the opportunity to advertise this community. And please come join us. Please find us on Twitter, find our website, and shoot an email to a leader to any working group you would like to join. And please come to the next workshop, especially since it's going to be a virtual. Hopefully more people can join than the in-person workshops. Thank you very much.
That was great, Kate. Isaac Ellman asked, is there somewhere online where I can read about the working groups?
Yes, Build a Cell has a website. It's buildacell.org. If I believe you can just Google Build a Cell, and after going through a few hits for the video game with a similar name, you can find us, and you can find a description of all the working groups.
Great. I have a question, which is any community sort of self-defines, and you've made that very clear. But in this meeting, I at least have seen some remarkable technology for analysis of synthetic cells. So let's say that folks from this community wanted to participate in a self-organizing effort within the context of Build a Cell. Let's say they wanted to set up an analytical group where they could share sort of results and approaches. How would that work?
They should set up their working group, ask me to put information on a website, and then if you build it, they will come. That's been the strategy of the Build a Cell from the very beginning. The only working groups that survived are the working groups that people came to and started working with. Since we don't have a central organization, all the working groups are completely organic. If you have an idea for an area that we're not touching right now, please set up a working group. I would be happy to advertise it to the Build a Cell mailing list. I'm used to spamming a lot of people that signed up for updates about Build a Cell, and you as the group leader define what kind of activities the group will have. So some groups have been meeting over calls between the workshops. Some groups have been working on shared Google documents. Some groups have their own Slack channels, as far as I know. I'm not invited to some of them, so I don't know what they're doing there. But basically, groups are self-organizing. And during the workshop, if a group has members, that group will happen. We basically vote with our feet at the beginning of every workshop and saying which working groups will happen at any given workshop. People sit at tables that belong to specific working groups, and that's how working groups happen. So basically, it's all kind of unstructured and organic.
Perfect. And so certainly, I think that all of the folks who participated in this conference have, their own ideas. I would encourage them to get in touch with Kate. I think this conference, especially the fact that it's going to continue into next year, is a great start on fomenting some ideas that would be community-wide. Similarly, for those of you who's participated in sort of the abiotic or in vitro biology groups, which have slightly different conferences. There's an obvious and interesting overlap. Oh, and I didn't even notice, apologies. There was a second question. Sebastian Lopez Castellanos asked, thank you for the nice introduction. We have heard yesterday and the day before about European initiatives such as Matter to Life and BASIC that also work on building synthetic cells. Has there also been an attempt to work together with those initiatives?
Yes, we're very lucky to have people that lead those initiatives to be members of Build a Cell. And we are looking for ways to kind of work, to kind of combine our working groups, combine our frameworks. One thing is the European initiatives are much more organized, much better organized, much more formal than the Build a Cell. For example, the community that Marilyn was talking about yesterday, they're funded, they're within one country, and they're actually they're geographically close to, so they can actually do research projects together. Build a Cell right now is only able to facilitate connections and facilitate interactions. We don't have research funding. The Build a Cell itself doesn't have any research funding. So all we can do is connect people so they can get their own research funding within their groups. So that's kind of all we're trying to do. And so the goal of Build a Cell is not to replace smaller initiatives, national initiatives like the BASIC or like FabriCell. The goal of Build a Cell is to build a network so organizations like that can communicate with each other, can connect better. And people that don't belong to any specific country organization, for example, if you're from a smaller country that doesn't have synthetic cell research, through Build a Cell, you should be able to join the international community.
Great. Danielle Rose asked, there is a buildacell.io and a buildacell.org, which is the correct website?
We just recently migrated to buildacell.org. So that's the one we're actively updating. The buildacell.io needs to be redirected to org. And it's because Build a Cell is such a grassroots informal organization, we were actually having a little bit of a hard time tracking down how to get back to the original registration of the IO domain and set up the redirect, but it's on top of to-do list of a person who can actually do it. So hopefully that will happen, but buildacell.org is the current one.
Great. So assuming there's no more questions, we're going to segue seamlessly to the final panel discussion. We're just going to power on through this. And so the final panel discussion is future synthetic cell technologies for mitigating viral pandemics. And I will admit this was sort of my brainchild. And so if it falls flat on its face, you know who to point at. But the idea was that, I'm associated with a variety of communities, as I said at the very outset, and I always try and challenge those communities to do something practical. And, you might think that Build a Cell is purely a theoretical, as it were, exercise. I mean, there's certainly experiments that go with this, but that, the goal, the grail is the synthetic cell. Along the way, what can we get out of that? So we've already heard an introduction for Kate. My own introduction would just be that I'm sort of a general purpose biotechnologist. With that, I would like Dr. Bodenschatz to maybe say a few words about himself.
Well, I'm Eberhard Bodenschatz. I'm from the Max Planck Institute for Dynamics and Self-Organization in Göttingen. I'm one of the founding members of the Matter to Life School. I'm in Max in Bio, and I work on fluid physics, cloud physics, but also on synthetic biology and cell biology.
And finally, Dr. Grunza, if you could do a brief self-introduction.
Professor,
yeah.
Can you hear me?
Yes.
Okay, I'm a physical chemist, and I'm a professor emeritus from the University of Heidelberg in Germany. And so now I'm actually, after my retirement from the duties of being an institute director and so on, I joined the Max Planck School Matter to Life, and I try to help where I can and also help with the teaching.
Okay, great. And I would encourage the audience to, as they have so far, put up questions that they find relevant. I'm going to pose to the panel sort of three-ish questions that have to do both with the current and with future viral pandemics. And they basically have to do with how do you make a virus or make proteins for a virus, how does that virus infect a cell, and how does that virus move between cells or even move systemically? And in all cases, can the synthetic cell technologies be helpful? And so in introducing that, I'll say for myself, one of the things we do in our lab right now, because we're trying to work on both antibody and vaccine development, is produce the spike protein of SARS-CoV-2. The spike protein is heavily glycosylated. It is huge and it is very difficult to produce. You produce it in slightly different cell lines, you can get slightly different answers in terms of glycosylation. What does the community, what do representatives of the community think are the possibilities for doing something like glycosylation synthetically or in a synthetic cell? Could we have a synthetic ER and a synthetic Golgi? How well can we control this, and how might we control this? And I'll encourage Kate to answer first.
Right now, we have excellent system from Mike Druitt's lab that allows for glycosidation in a bacterial cell-free system. And we have those old canine microsomal membrane and similar systems that allow glycosidation using other protein expression systems. I think to actually build a cell, we have to figure out the control problem. As you mentioned, Andy, it's the biggest thing is how to control glycosidation. Not just how to do it, because we have enzymes, and from the recent Mike Druitt's work, we have really well-working enzymes that can do glycosidation. The biggest problem is how to selectively control it. I don't know how to do it. If I knew how to do it, I would love to use it in some of my systems that are in a desperate need of selective glycosidation. But this is one of the areas that the field is developing really fast. We know that for both building a functional cell and for biotechnological applications, we need to solve efficiency and control of glycosidations, and we're getting really close to that.
Dr. Bodenschatz or Grunz, any comments?
Well, I just like to remind you about the tutorial we had by Reinhard Lipovsky at the beginning. And he was talking about membrane physics, and also he was talking about the endocytose of particles into a giant vesicle. So I'm not sure. I mean, not to make it complicated, but by more or less physical means, maybe it is possible to functionalize vesicles with the receptor proteins, the AC2, and scavenge, so to speak, the virus before it actually enters the lung and the other organs. I just read something very interesting today, actually, that there was a study going on that you can actively, or you can apparently, I should say, reduce the concentration of virus in your mouth and in your nose by using an antiseptic. And a fairly strong antiseptic, but maybe you can make a sort of a foam or barrier of artificial cells with the right receptor molecules to scavenge virus before they actually get into your organs.
That is an absolutely brilliant answer to the second and third questions that I was sort of going to pose, which is to the extent that we can make, let's say, appropriately glycosylated proteins or even a viral capsid, how could we examine its infectivity? And you point out from earlier in the conference that we can potentially look in a synthetic cell or a synthetic cell-like vehicle at uptake and endocytosis, and then perhaps we can look at issues relating to viral transport within a system, such as barrier functions, the inclusion of barrier functions as a therapeutic. I'm also reminded, relative to some of the things Kate was saying, that earlier today, we saw a very interesting talk from Nick Carroll about, essentially segregating proteins into their own compartments. And I was curious whether, we may or may not be able to recapitulate the Golgi as a membrane-bound apparatus, but could we have enzymes that would self-separate, that would do glycosylation in a particular order in order to efficiently make proteins? And would that potentially, especially given the possibilities of in vitro transcription translation that we've heard about within encapsulations, lead to programming? So I think that we all sort of see on the horizon these opportunities, but as Kate pointed out, they're going to be really hard. it's going to be really difficult to get there. Dr. Bodenschatz, do you have any comments here?
Well, I wanted to share this, which I shared a moment ago. This would make clear a little bit what you were talking about, right? If I understand this correctly. And so there seemed to be, the one thing that I found especially interesting, which goes to all the other answers that you've already heard. One of the challenges just going through the plasma membrane and seeing how does this thing actually dock into the cell. There are two pathways, right? One is endocytosis and the other one is actually merging over the membrane, and it just goes inside. And so these are these two pathways. One's the receptor mediated and through the endosome, and the other one is going through more just a direct way where it just docks with the membrane. This is something that one could rebuild, perhaps, by making basically vesicles that have the right receptors. I don't know whether it's possible. One could think about it. And then just looking at the entryway. I think this is also what Michael here was talking about, right? And we wanted to interfere with this. Then the big challenge to me is down here. This is the part where I do not know how to build this at all. How do you make these membranes? How do you make these large surface areas in an artificial cell that we need to do this? These are not simple vesicles. This is a very complicated structure, which is a highly membrane structure, which has an enormous surface area. And I would love to see a microfluidic system or some system where we actually can incorporate some lamellar structure that has a membrane, which is functionalized, even enzymatically active, to have something like an artificial ER. This would be wonderful. I don't know of any attempts to actually doing this, but I think once we understand this, then we can actually start using these little cell-like objects as machines that produce things for us. Because right now, the turnaround, the efficiency of these systems that we're building is just very low. Mm-hmm. So if you want to get energy in these systems, we have to play the game nature using, which is putting in membranes and large surface areas. Thanks. I will stop sharing this. I think you have seen it. So thank you.
Yeah, I think one of the reasons this is an interesting question for me is that folks who actually try and work with the live virus, and this will be true in future epidemics as well, are frequently confronted with fairly heavy-duty biosafety restrictions, as they should be, right? So it's BSL-3 for us to work with this virus. But but that's because, of course, if the virus can propagate, then the virus can potentially get out of the lab and infect people. I think that as we build synthetic systems that cannot allow propagation, but allow the dissection of different properties of the virus, of glycosylation, of entry, of potential passage through barrier functions, that we may be able to create a much better biosafety environment using synthetic cells. And the equivalence of this in synthetic biology is, while folks in some ways are just beginning to realize some of the great benefits of synthetic biology, there was an intermediate stage where synthetic biology was thought to be a great means of controlling how hazardous things might get made. You'd have unnatural genetic codes or unnatural genetic alphabets. And similarly, I think we could have unnatural cells, synthetic cells, that would allow us to examine viral function, but without the attendant hazards of worrying about whether the virus was going to replicate. And so that's what I wanted to set out as sort of a challenge to this community, although I totally agree with you, building a synthetic ER or a synthetic Golgi looks really hard. But is there something short of that that would still be useful that we could attempt to build?
Yeah, well, I think, the nice thing about bottom-up synthetic biology, or this type of synthetic biology we're talking about, is that there's hope that we can build some relatively simple systems, like you were just describing, that allow us to be really quantitative about the processes that we're studying by reducing the elements that we actually put into the system and seeing whether the things we conjecture, a lot of in biology, a lot of things are conjectured. You basically say, I've done this and this and this and this, and because of this, it must be like that. Perhaps this is a way actually to show that it is actually like this, and you can get away with a few components. You don't need the whole cell. You don't need the whole complicated complexity of the whole cell. You can actually get away with these simple ideas that we as scientists usually have. And probably most of the time we will be falsified. Most of the time we will find out, no, it doesn't work. But I think this bottom-up approach where you do simpler systems is actually a really good way of doing it. I don't think we can get rid of the class 3 laboratory, because probably still a human is going to be in the room. And the best replicator for a virus is so far probably the human. And so I wish, we could do this, but as long as there's a person in the room, you probably still need a class 3 laboratory.
Absolutely. I think one of the other potential advantages of these synthetic systems is, again, I've been impressed throughout the conference with the analytical methodologies that have been brought to bear. Actually, being able to test those analytical methodologies out or proof them on simpler systems and bring along the quantitative sort of metrics for how they work and under what conditions they work would potentially be a stepping stone to working in cells, working in tissues, even though that's, of course, where you really want to potentially use these. Nonetheless, the methodologies themselves could potentially be worked out on these simpler systems. And that's also, Kate, honestly, why I'm very excited about there being potentially an analytical group in Build-A-Cell. Also, you don't actually have to build everything at once. The nice thing about building cells is that because they're already, they're dead, so they don't care. You can divide functionalities between different kinds of cells. So even if we can't build a very complex internal structures like Golgi, we could theoretically outsource activities of different parts of that system to different kinds of synthetic cells. I don't know how to do it again, but one single synthetic cell doesn't have to do everything. You could try to do kind of distributed computing model. I'm not saying that's going to be easy. Yeah, it's a good idea, and even practically, as I believe Dr. Grinza was mentioning, let's say you had variants of an ACE2 receptor or variants of a viral spike protein, and you distributed those to different vesicles, different cells, and you wanted to just see, in an evolutionary sense, how a pseudovirus, not a replicating version, would assort itself, right? Library on library sorts of things. That's not a trivial thing to do. And doing it safely is really difficult. But nonetheless, it's a very interesting question in terms of looking at the forward evolution of the virus. Okay, do we have any thoughts, any final-ish thoughts? I don't have any questions up right now. People are more than welcome to ask questions from Dr. Bodenschatz, Dr. Grunza. We've talked about some of the questions that I'd thought about, but I'm perfectly happy to let you bring up additional issues.
I'm just very optimistic that all these activities around the world will be successful helping us in controlling disease. And it's amazing how many people come together. We more or less listen to the biophysics and biochemistry community, but there's a lot of activity also going on at the large scale installations like the X-ray laser in Hamburg and synchrotrons, where there's a very efficient way now to protein crystallography, and also with these XFELs, which are coming online, where you can do really just shoot a X-ray beam at one cell, you drop them through a cavity, and then you can reconstruct the whole cell in a very reasonable time. So I think the analytics we are developing and outside this community we just came together are amazing, and I think we should tap on that a little bit more to speed up our development work.
Dr. Bodenschatz?
Well, I'm really excited about the opportunity of building these systems and really being quantitative, both experimentally, but also theoretically. So the real look at complex systems, complex system interaction, different reaction diffusion systems that interact with each other and actually make the function of what we call a cell. And to really decipher the individual elements and systems and really understanding them, how they interact. I think this is very, very hard to do in biology, because biology has produced in cells so many different functions and always escape routes. You knock out one system and sure enough, there's a second one who does the same thing. And because the cell has this enormous genetic code where it just says, let's make a few other proteins and enzymes that will fix the problem. And this is because of evolution, while our bottom-up systems don't have that evolution yet. And so we can really hopefully put in what we need and do real quantitative tests and then make some actually quantitative theories. So for physicists, this is a dream come true, I hope.
Great. Dr. Adamala, any last thoughts?
I just want to build a cell. That's my biggest goal. I think what we build is going to be much simpler than a natural system, obviously. But I am really looking forward to seeing some emergent properties and building the complexity up. And then, as you said, we need to find ways to analyze it. And then we're going to learn a lot about those fake cells, but also about how natural cells realize those functions.
Great. We have a question from the audience. Ravi Ram asked, after the viral fusement, attachment shared had nice graphic, is it known how many copies does the virus make of itself from an infected cell? Does it drive to make more membrane components or sort of melts, breaks, and uses what the cell already had? Nominally, I should probably know something about this, but I don't. And so I actually don't know how many viruses are made per cell, nor whether it redirects the cellular membrane material. I suspect the latter, it doesn't. The viral replication cycle is relatively fast, and so the actual transformation of cellular metabolism is probably not going to be attendant on the viral life cycle. But I don't know what the yield is per cell, more than one, and it may differ on a cell-by-cell basis. And we know this virus can go to a variety of cell types. So I can't give you a really good question there, Ravi, but maybe my colleagues know more than I do.
I mean, one of the tests of seeing whether the virus is active or not is to put them on cells, right, and to see how many cells are actually dying. So most likely it would be efficient. I don't actually know the number, how many, but the cells die, and otherwise the test wouldn't work, right? And so I don't know, but clearly it's not good for the cells. Otherwise we wouldn't have these tests. So I can only.
Can you imagine to put a cell into a giant vesicle and then infect the vesicle and study the virus cell interaction, just in a confinement, in order to answer these questions? What is the cycle and how many viruses are being produced?
Absolutely, except I think I do it the other way around. One of the things that we and others work on, of course, is emulsions. And so a water and oil emulsion with a cell inside is sort of a perfect isolated test tube. And doing emulsion-based experiments is one way to look at large libraries, many events, and so forth. But a real problem with emulsion-based experiments is that they tend to not work with large cells. They tend to not work with complex cells. It's very difficult to do eukaryotic cells in emulsions for a variety of applications. So having a simpler system that would be better encapsulated would be of incredible utility. And as Kate points out, even if there's just a subsystem there that you can look at, it might nonetheless be a good way to, on a synthetic cell by synthetic cell basis. Because another of the questions I sent out was, could we envision sort of a digital technology for looking at virus production or virus detection on a cell by cell or emulsion bubble by emulsion bubble basis? But I think it's a really good thought. I think I just sort of switch it around in terms of what was the outside and what was the inside compartment.
Okay, yeah.
So I like the idea actually from just very well. So what you could do is you could make an artificial tissue where you have a lot of synthetic cells, which are not really cells, they're just vesicles. And in between there's a few cells that are actually just having neighbors and stuff like that. And then you could infect that tissue. And then this way you have an isolated cell being infected by the virus. So you don't have to make a compartment for the cell. They put a lot of other compartments which are not taking part next door. And this way they would be isolated. So I like the idea a lot. I mean, this is a way to really do a single cell experiment by having all the other cells far away, hopefully.
Yes.
Well, exactly. And that's one of the reasons I was excited about Dr. Laporte's talk, Delaporte's talk earlier today, and some of the earlier talks that were, somewhat more on the biology side, because they basically began this process of dissecting functional tissues into their simpler components. And so in all ways, in the synthetic biology community, we're always thinking about dissection into rational components and then rebuilding. But for cells, that's really hard. I mean, Kate points out how very hard that is. But to the extent that we can take that problem on, we can take it on up to the multicellular level and then potentially even beyond. When we think about barriers, they don't have to be barriers inside a cell. They could be barriers at the sort of surface of a tissue. They could be barriers within particular tissues. And what would the physics of those be like for viral transport and viral transfer? Those are hard questions, but they may only be questions that we can realistically answer with reconstituted systems from synthetic cells. Okay, sorry, Ravi has a second question, apologies. So virus is a type of minimal cell, just lacking replication and metabolism. Thanks a lot for the excellent symposium. Overall, awesome initiative, build a cell. So I think that was more a comment than a question. And that's a perfect sort of comment to potentially go out on. I'd like to thank my fellow panelists for taking part in this exercise. I learned a lot, which is, sort of a selfish thing, but I think it was very great to hear your perspectives. And with that, I'm going to turn it back over to Gabriel, I believe, for closing out the conference. Thank you all so very much.
Thank you. Thank you.
Thank you.
Well, that was a really fantastic session. I am very grateful to all of the participants, and all of the speakers and panelists today, and over the last couple of days. I think this has been a huge success. Could I have the slides, please? So this is the, I think, just the first of a series of conferences that we will be having over the next few years, where we bring together people from across the globe to discuss this quest for building synthetic cells from the bottom up. So this, I think we can count sort of as the inaugural international conference on engineering synthetic cells and organelles. And I think there is plenty of room to have more conferences going into the future. And given the degree of interest, I think this will make good sense. So again, thank you to all of the experts who gave their time to help make this a success. And thank you for all the participants who have joined and asked some great questions along the way. Next slide, please. I just have a few concluding remarks. This slide shows or indicates the reach of our virtual conference. It's truly an international conference. We've had over 700 people participate from 47 countries around the world. Over 200 different institutions were represented among our attendees. Around 80% of you are early career researchers, so we're really excited about the impact of this meeting, the growth of this field of bottom-up synthetic biology, and the future of the SynCell network. For this, I want to thank everyone again, and I want to make sure that we keep the conversation going. Feel free to use the hashtag SynCell2020 on Twitter. Next slide, please. I once again would like to thank folks who did a lot of preparatory work for this event, and especially folks at UNM and Max Planck Matter to Life, the UNM staff who have been working on this for several months, the organizing committee, and especially my co-chair, Dr. Joachim Spatz, and then again, the speakers, panelists, and moderators. I also like to acknowledge partners who have provided resources to enable this conference and our subsequent conference, NSF, the University of New Mexico, Max Planck School Matter to Life, Los Alamos Laboratory Center for Nonlinear Studies, and the New Mexico Consortium. Next slide, please. Special thanks to our technology team at the Max Planck Institute for Medical Research, and Dr. Andrew Holle and Dr. Jennifer Young. They have been extremely professional and very cheerful throughout all of this, putting up with some and solving problems that arise along the way, always with good cheer. I really appreciate that. And I think that the idea that these young folks had to have this virtual conference in lieu of our in-person conference this year was brilliant. I think this has gone along very, very, very well. Thank you to both Andrew and Jennifer. Next slide, please. I just want to remind you to keep your eyes on SynCell2020.unm.edu. We will be planning over the next year for in-person conference. There may be a virtual component to that. We shall see how things develop. Next slide, please. So far, the plan is to have this in Santa Fe, New Mexico, and I hope you will all consider to come visit us in New Mexico. We welcome feedback and suggestions regarding speakers for next year's conference. Turns out that this email address here, SynCell2021@unm.edu, is not quite active yet. It will be active in the coming days, but in the meantime, you can just use SynCell2020@unm.edu. We welcome all ideas for next year. Next slide, please. Okay, so finally, we would like to try to generate a group photo, a virtual group photo for our conference. Perhaps I could have Jennifer come on, and you can actually see the amazing Jennifer and Andrew if he's willing. I have actually some questions about this. The idea basically is to generate a sort of collage that we can use on our websites and in our future garnering of resources for this type of event. Basically, my question for Jennifer and Andrew is whether these times will still hold, given that it's only about 11:20 right now. Basically, what we're going to have to do is, because the webinar format in Zoom does not allow all the participants to use their cameras, what we would like to do is log off the webinar and restart on a regular Zoom conference at the times indicated, and then everyone will be able to share their photograph. Because Zoom only allows 100 people at a time on a regular Zoom meeting, we thought we might have to do this a few times in order to get everyone. Jennifer or Andrew, shall we stick with the times indicated?
I think we can bump it up 10 minutes or so. So
there they are. There's the round of applause for Jennifer and Andrew. So, would you like to start then at 11:30 Mountain Time, 7:30 Central European Time?
Yeah, we'll bump it 10 minutes. I'll start it right after this.
All right. So I very much encourage all of our participants to try and to get into one of these sessions. It'll be basically every 5 minutes starting at 11:30/7:30. And we would really like to get as many people as possible on our collage. I think, as I mentioned earlier, especially important to highlight the youth of the crowd, and also the diversity of the crowd and the geographic diversity as well. So I think that is it. Jennifer, Andrew, is there any last instructions you would like to provide?
Yeah, just that the link for this this photo session is in the chat right now, but it's also in the email that everyone should have received already.
And that chat went to everyone?
Yes.
Okay, fantastic. So all you have to do is copy that link and join at one of the times on every 5 minutes, starting at 11:30 or 7:30, or wherever you are on the half hour. All right, well, with that, thanks again to everyone for your participation. We hope to see you in Santa Fe next year. Bye-bye.